All right. Good morning, good afternoon, and good evening, everybody. Welcome to the Analysis Working Group Symposium 2022. Before we begin, I just want to give a big shout out and thank you to KBR for hosting this platform, for providing the platform and hosting the event, and for Marco Bolt for his stellar support for the AWG Symposium. Before we begin, I just want to cover two really quick housekeeping items. And the first one is that we will be putting out putting a link in the chat, which Mark will put the link to in the chat for this sign up form for the analysis working group. If you are interested in joining, if you're really interested in the science that's happening and would like to join, then please fill in your information in the sign up form. And we will be putting this link in the chat throughout the event. So if you happen to miss it the first time, don't worry. The link will be in the chat throughout the event. And lastly, if you have any questions, if the attendees have any questions for the speakers about their specific presentation, then we'll be using Conference.io. And for those that are new to Conference.io, this is what the platform will look like. And you can click on the plus sign here to be able to add a question. And then once you type in your question and submit, it will be public to anybody that has that link open. You can use the caret here to upvote any questions that you may like. And I will try to do my best to answer as many questions as prioritized. I will most likely be answering questions that have the highest number of upvotes. So with that, I would like to hand the reins over to Dr. Jonathan Galazka, the GeneLab project scientist from NASA Ames Research Center.
Great, thanks, Lavorka. Before I begin, I really want to recognize Lavorka and thank her for her work on this symposium. She's done an enormous amount of work organizing this and has done a really great job. And of course, pre-thank all of our wonderful working group members who all volunteer their expertise to drive our science forward. And with that, I'll give you a bit of a background on space biology, open science, GeneLab, and these working groups. Next slide, Lavorka, please. So space biology is dedicated to understanding how life responds to the spaceflight environment. And it's organized into 5 basic elements: plants, animals, developing organisms, microbes, cells, and molecules. And in each case, we want to understand this both because it has applications towards making human space travel safer and more effective, but also because there were fundamental questions that we would like to answer using the space environment as a platform. Next slide. A major recent effort, is to now leverage open science tools and platforms to accelerate the pace of discovery in space biology. And we're going to highlight four efforts here. One is the Biospecimen Sharing Program, which has a dedicated team which helps to dissect and collect excess tissues, which can then be deposited into the NASA Biological Institutional Science Collection, NBISC, which is an archive which allows public access of these spaceflight tissues. There's the Ames Life Sciences Data Archive, which houses sort of more general data, mission data, and phenotypic data from space biology experiments. Then finally, GeneLab, which is dedicated specifically to the housing and curation of omics data, so large-scale data. GeneLab, next slide. GeneLab now houses 378 studies, which contain 430 data sets. And these data sets span all of the model organisms that are used for space biology research, so that's now 45 species, and GeneLab also accepts any assay type, so omic scale assay type. Most of this is transcriptional profiling because that tends to be what the community uses, but protein expression profiling, DNA methylation profiling, those data sets are housed within GeneLab as well. And we now have over 130 terabytes of data. Next slide, please. And GeneLab has a number of important functions that allow us to maximize the value of these experiments. This includes the ability to discover this data. It also includes tools for directly exploring the data, so a visualization portal that allows exploration of these data sets without having to download and analyze the raw data yourself. It does include tools as well for doing de novo analysis of the raw data through the Galaxy platform. And then upstream of this now is a portal for the contribution of new data from space biology PIs. Next slide, please. So, and advance one more because there's an animation. All of these tools have shown allow GeneLab to be an excellent resource for educators and students, and we have a variety of programs that allow students to engage with this data. One is GeneLab for high schools, another is GeneLab for colleges and universities, or GL4U, and then the SLSTP internship program, which oftentimes utilizes these tools as well. Next slide. Next slide, Lavorka. Oh.
Okay,
so that's,
sorry.
while Lavorka brings those up, that brings us to the analysis working groups. And these groups were started in 2018. And the motivation for this was to develop a community, intentionally develop communities dedicated to utilizing this data. So we started these groups, and we now have them broken up into 4 major foci: microbes, multi-omics, animals, and plants. And there's cross-pollination between those groups. And these are groups which are volunteer groups, and they are dedicated to, so here are 4 groups, and they're dedicated to looking at this GeneLab data. And ultimately, so we can go to the next slide. So those are the 4 groups. We have a 5th group, which is the Ames Life Sciences Data Archive Analysis Working Group. This working group is the newest and has already been very productive, and is looking at ways that we can systematically bring in new data types into the ALSDA. The next slide. So again, the purpose of these groups is there's multiple purposes. One of which, the primary goal of these groups is typically to publish new investigations. So using the open science resources on GeneLab to make new discoveries. But along the way, this provides a lot of value back to GeneLab. This critiques our curation techniques, our processing techniques, et cetera. So they feed back pipelines and suggestions for reorganization, et cetera. Next slide. So what are the expectations and what are the benefits of participating? The expectations really are to contribute. So to come to the monthly meetings, to contribute your expertise, and this can be at a variety of levels, both bioinformatics expertise, but also biological expertise, project management expertise. All of this is welcome into these groups. And the benefits, there's a lot of benefits: networking, learning from the experts in these groups, and then just advance to the next animation, and ultimately the publications. And so we've already had a variety of high-profile publications coming out of this group, one of which went into Cell, and so it's a great opportunity to get on some really nice publications and to learn a lot from some expert scientists, including experts on space biology, which is a field that you wouldn't necessarily run across in a typical PhD. Next slide. And so that brings us to today. So this symposium is meant to highlight all of the great work that is going on in these groups, and to hopefully not only show you what we're doing, but also maybe entice new members into these groups. So we're really always looking for new members with new expertise and perspectives to help these groups move forward. And so with that, there's one more slide, so I'm not going to go through everyone here, but we have a variety of AWG members that are going to tell you about their work, and I'm really excited to have you see that, and I'll turn it back to Lavorka.
Thank you so much, John. That was a great synopsis of what it is that GeneLab and the AWGs do. And right now, our next speaker is Dr. Kate Rubins, and I'm absolutely honored to introduce Dr. Kate Rubins as our keynote speaker. She was selected by NASA in 2009 and completed her first spaceflight in 2016, where she became the first person to sequence DNA in space. She holds a Bachelor's of Science in Molecular Biology from UC San Diego and a PhD in Cancer Biology from Stanford University Medical School. After her PhD, she worked as a principal investigator at the Wyatt Institute for Biomedical Research and headed 14 researchers studying viral diseases that primarily affect Central and West Africa. Dr. Rubins most recently served aboard the International Space Station as a flight engineer in 2020. And across her 2 flights, she spent a total of 300 days in space, the 4th most days in space by a US female astronaut. And today, Dr. Rubins will deliver the keynote address titled Conducting Research in Microgravity, a Scientist Astronaut's Perspective. Welcome, Dr. Rubins. It's a pleasure to have you, and please take it away.
Thank you so much, Lavorka. I'm really excited to talk to all of you. We give a lot of talks at NASA, but it's rare that I get to speak to an audience of scientists. So this is going to be really exciting. I will share my screen now and get started. I'm going to show a short video first before we get really going. I wanted to kind of show you a little bit about what it's like to conduct research on the International Space Station, and then talk about some of the experiments that we did to cover the breadth of things. So if you guys have Lavorka or Marco, if you have the video up, we can go ahead and start that. So this is about my most recent flight, Expedition 64 to the International Space Station. Training for this really starts about 2.5 years before we actually launch. So I'll fast forward through most of that and get right to launch day. I launched out of Baikonur, Kazakhstan on a Soyuz. This was my 2nd time doing that. And launch day is pretty exciting. You've trained for this, for the last 2 years, but really your whole professional career, essentially. It is absolutely an incredible experience to lift off in a rocket, to know that you are within 9 minutes going to be going 17,500 miles an hour and on your way to the International Space Station. It's a pretty big achievement to reach orbit and have all the systems working nominally, and then get to our destination, which is the space station. This was a very short trip for us. It was only 2 orbits until we rendezvoused with the space station. This was my 2nd time being on the space station, so I was pretty excited to see some crewmates that I hadn't seen in a while, and to get together with everybody that was on board. Once we get there, we get really busy right away, starting in on the work. And so usually we participate in about 200 different experiments. We can either be the participants as human subjects, or we can be conducting the experiments. So you'll see a little bit here. This is a plant experiment that we actually, you take a look at the entire process of the plant development, and then harvest the plants for sending this home for later analysis. There's not just biological sciences, but there's a lot of physical and chemical sciences. This is a combustion experiment. We are studying different properties of combustion in space, and we have found a lot that's very different in space about the properties of combustion. This is an experiment to look at free-flying satellites, and so we actually can have these satellites remote controlled from the ground, either by researchers or through student groups. This is run out of Ames. There's a lot of experiments that are looking at fluidics. Fluidics completely changes in microgravity. And so this one was investigating how plant roots might grow in a fluidics environment. Of course, we're doing a lot more microscopy now. This is really in the last 5-7 years a newer thing for space station. We're able to do a lot more tissue culture than we were before, and we're starting to do many more long duration tissue culture experiments, which I think is incredibly exciting. It paves the way for these multi-generational development types of experiments we'd like to see in the future. We're usually always working in these kinds of glove boxes here, both to protect the cells and to protect the astronaut from our experiments, since we live and work in our lab at the same time. And these are incredibly popular for the astronauts. I'll talk a little bit more about this heart cells experiment. But people really, whether they're from a scientist background or not, they really enjoy interacting with the science. Anytime you can see something or watch a process go through on board, I would get crew members crowding around to see what we were doing. I'll talk a little bit more about some of the DNA sequencing experiments we did on board, but you can see this is actually putting things into the sequencer. We also had video microscopy for capturing genetics and genomics experiments to look at nematodes, to look at both structure and function. And lastly, I really wanted to say, you know, sometimes we're working in space, a lot of things are improvised. So this is a centrifuge in space. We have other centrifuges, but this is a 3D printed rotor that we put on a drill, and it actually works very well to get your samples down to the bottom of the tube. A lot of times we're using things like duct tape to keep our samples in place. We have pretty high-tech interaction with the computer systems on board and the ability to send a lot of data down, but we can't always send those samples down. That volume is very limited. So the more analysis we can do in situ on board the space station, I think is actually really important, and that can really help researchers. This was kind of a personal fun experiment of mine. I really wanted to see how we could handle fluidics and microgravity using standard lab tools. So the question is, do we always have to build special containers or very fancy machines to do our payloads? And it turns out that the really small well volume of these 384 or 96 well plates contains the fluid quite well. We generally fill it to half or less full, so that we don't get well-to-well contamination. But there's so much surface tension and adhesion forces, it really keeps the fluids in the plate. And so we're starting to expand that thought process. So we had a ton of fun and 200 experiments, but you know, at some point the mission has to end. It was pretty sad this day. We got in our capsule, we undock, and we just do one orbit around the Earth in our tiny little Soyuz. And then we light the deorbit engine. This is an interesting process. You're going through a fireball, decelerating from 17,000 miles an hour. So it's a good feeling when the parachutes come out. What happens next is rather exciting. It's an Earth accident. You slam into the planet and then greet the SAR forces that are there to rescue you. So you're pretty much just genuinely glad to be alive at this point. I told all of them they were my best friends in the world. I was pretty excited to see people again after 6 months and to see this great team that supports all of our spaceflight adventures. So with that, I can take over the screen sharing and start the presentation. So I showed you just a little bit because I think video is fun to see how these experiments look. And I wanted to just talk about how we do experiments on board. So some of the things that we're looking at, sorry, that's me. Some of the things that we're looking at are really driven towards understanding the human response. And we've got two reasons for doing this. We want to see how astronauts can perform when we start sending them really far away from home as we go into deep space and to Mars. And we also have a lot of disease parallels in spaceflight that we think can reflect diseases on Earth. So for example, neurovestibular, you have very strange neurovestibular alterations. This is a huge problem for balance disorders on Earth. Cardiovascular, we're really starting to understand that this may be more of an issue. There's a lot of study that we need here. Bone, we've understood for a long time and have very good countermeasures for, but we have a lot of muscle wasting, even with all the exercise we do, 2.5 hours a day. That's still an issue. We know that the immune system is moderately dysfunctional on board. we're really thinking about nutrition as it relates to long duration missions, where we might have to send the food many years ahead of the actual astronauts who are then going to be, on this 2.5, 3-year journey. Certainly, as you go farther into deep space, we're thinking about how the crews are going to interact, how they're going to interact with the ground, make sure we have a really high functioning, high performing team the whole time. And then lastly, radiation is an issue in low Earth orbit, but as we leave the protective magnetosphere, we're going to have much more serious issues with radiation. And so all of these are really interesting areas that we're studying now and in the future. Just to give you a sense from the human perspective, we're in homeostasis on the Earth. We suddenly launch people to outer space. We throw them into this environment that humans have not evolved to be in. And we have a lot of things going on physiologically in that first week or two. And so you can see the neurovestibular system is reacting like crazy. Your fluids are really having a big issue because you don't have that pull of the fluids down to your lower half of your body. Your cardiovascular system is adapting. We get changes in our red blood cell mass and in our plasma volume. These things, after a few weeks, really kind of get into a homeostasis. And so a lot of times we talk about the human body and all the issues that we have. Some things are issues for long-term health in space, but some things are just the human adapting to homeostasis and microgravity. Our bodies are so incredibly adaptive, and we're getting good at living in microgravity. Use it or lose it. We don't need that bone structure anymore. What happens is that we become maladapted to living in 1G. And so those are the health issues that we're really worried about. So we're not worried about trying to increase the plasma level on board because we actually have a very good plasma volume. Where we have to really think about it is when we're coming home to land and all of a sudden you're going to be massively dehydrated. So we're also thinking about these things like bone loss and radiation loss that are cumulative, that don't necessarily steady state homeostasis is not what's going to happen there. You're going to maybe get continuous loss as you go on longer and longer duration missions. So today, tomorrow, and the future, today is ISS. We've got these 6 months to 1 year missions. We've got real-time communication with mission control. We can return samples frozen. We can evacuate the crew members if they have a medical issue on board. As we get out to these Mars transits, we're now talking about up to 900-day missions. We have a long communication delay. We've got much more limited mass and power and volume than we do on the International Space Station, and we can't evacuate the crew. So we're going to have to think about human health a little bit differently as we head to Mars. So your lab today is the International Space Station. It is one of the most incredible laboratories I've ever worked. The view is really second to none, but working in microgravity is a very incredible experience. And particularly as a scientist, the opportunity to use gravity as a variable. This is really hard to do on Earth. We can do it in short second bursts if we're looking at parabolic flights, or we can get some longer 4-minute suborbital. But if you want to study a process that's longer than on the order of a few minutes, microgravity is really the only place right now we can do it. It doesn't always look like your standard laboratory. So you saw that bench top where I had my pipettes kind of stuck on some duct tape and they were floating around. Sometimes our bench is just stuck on the side of the rack that has a life support system behind it. The whole space station kind of looks like this. It's sort of a hot mess. There's cables everywhere. And we are living inside both our HVAC system and our laboratory. We have some really good ways to do sample processing. So I mentioned the glove boxes, and that allows us to do things like sterile technique. We can do tissue culture in there. I've cultured a number of things for 30 days without contaminating them. And my students would tell you I'm not like the best at cell culture. So these are pretty impressive facilities. We have freezers on board the International Space Station. And so this doesn't look like your typical -80. It's in the floor and you pull it out and the freezer shelves float. But we do have space to freeze things, and we have down mass allocation to send them back to the Earth. Some of our challenges in microgravity are things like fluid dynamics. So this adhesion force is completely different. It's the same adhesion force that you would see on the ground, but because you're taking gravity away, it seems to dominate everything. We have bubbles that get introduced, and once they're introduced into your system, unless you have an active filtering mechanism, they stay there. Sometimes they break up and get smaller, sometimes they move around. But this is quite a challenge for molecular biology if you all of a sudden have unexpected air in the system. And so we think about that. But some of these things that cause us challenges, like the bubbles and the fluidics, are part of these properties of microgravity. And they're things that we actually want to study. This is part of what makes microgravity so interesting for biology. So cell culture is really a huge way that we want to take a look at this. This is the initial heart cells experiment that I did on Space Station in 2016. And we had these little 6-well vessels right here, which were designed to be like a 6-well plate, but injection ports to change out the media. We had a video microscopy system, and then we had the ability to take samples for RNA for later genomic analysis. This is the kind of the general overview. We used human cells from 3 individuals, took PBMCs, then reprogrammed these to be induced pluripotent stem cells, differentiated them into cardiomyocytes, and then sent half to the station and half on the ground. And so these were replicates of 3 donors. What we could do on station was look at these cells with video microscopy, and we could then also send them back to the ground for staining and take samples for genomic analysis. So interestingly, we did not see really huge changes in the cell structure. This was done post-flight. So we had already returned them to the ground and they were back in gravity. We didn't have the opportunity at the time or the facilities to stain the cells and use immunofluorescence on board, but the morphology was very different on board. And so I think this would be really interesting to look at immunofluorescence of cells along with their genomic profiles and some, tagged structural studies during a time course of cells in flight. And we're starting to have the immunofluorescence and the confocal microscopes that you would want to use for those experiments on board so we can do these analyses in real time. We looked at things like the contraction and the calcium handling. This was slightly altered in the space samples versus the ground samples. But where we really saw the difference was in the expression profiles. So this is RNA expression profiles. And we were looking at these samples, flight samples, post-flight or ground. And you can see that even though these were from different donors, the samples really clustered by the phase of flight that they were in. So the flight samples clustered very distinctly from the ground and the post-flight samples. Within those sample groups, the ones from individuals clustered tightly together, as you would expect, being from different human profiles. And what we really saw when we looked at the enrichment scores is a strong enrichment for genes that are in affecting mitochondria and energy pathways. We saw some interesting regulation of things like DNA helicase and DNA repair enzymes. And so this is really warrants further looking into what's going on with these pathways. How are these gene expression changes affecting the structural changes, certainly that we see in flight, and the function changes about the heart cells beating. So this paper is published. You can go take a look at that. And fast forward 5 years to 2021, I was lucky enough to fly again, and I was really lucky enough that I got version 2 of this experiment on board. So this was a very similar experiment, but instead of just looking at cardiomyocytes, we were looking at engineered heart tissues. So we were actually looking at both the cardiomyocytes as well as the endothelial cells and the cardiac fibroblasts. Here we decided to use chambers and suspend them between posts. And so you can see this is the engineered heart tissue suspended between 2 posts. So it has 0 interaction with the plastic. Very similar experimental design. We had all of these individuals. We took replicates of them, we put them in the plates, sent half to the space station, and did the same microscopy and the genomic analysis. We sent live samples back to Earth and then analyzed them on the ground, as well as the video microscopy and RNA samples taken from on board. So this is just a little bit, anybody who's launched something to space knows that you get a very cool side benefit, and that you often get to go see the launch. This is the team preparing all of these beautiful chambers. It's always fun to play a launch video, so I kind of have to do that. They came up to me on the space station. I was really excited to unpack these from the Dragon, put them in the incubator, and then took care of them for 30 days. So the media changes are very interesting on board. We're working towards things like open plate cell culture, but this was a closed chamber where we had to transfer the media through syringes and an 18-gauge needle. So this was an incredibly laborious way of changing the media. I'd love to develop techniques for this where we can go a lot faster because it cuts down on the crew time. This is the best that we could think of at the time. And it would take me 20 minutes to half an hour to change the media on a single chamber versus on the ground, 2 minutes per plate and you're done. We then got a chance to look at them via microscopy, and you can see this is actual microscopy images from on board, which then that data can be sent down and all kinds of analysis can be performed on this. You could definitely see some differences in the same samples from the same donor. You can see here the ground contractions versus the flight contractions were much stronger and occasionally more frequent. So we saw this really forceful contraction in the flight tissue. We could actually demonstrate this based on the data from the video microscopy, looking throughout the entire time course and seeing really the stronger contractions in all of these flight samples with our standard, demonstration that these are really significant. So that study is not published yet, but hopefully coming soon. I wanted to switch a little bit and also talk about sequencing. So we are doing a lot of DNA and RNA sequencing on board. I got to be the first person to sequence in space. This was really cool. I was so excited about this experiment. I'd worked with the PIs a lot. And so we demonstrated that it worked. We've got a lot of uses for this. We could use it for real-time gene expression. We could use it for monitoring clinical analysis of the humans during spaceflight, their medical health with diagnostic tests. We can use it for monitoring cell culture experiments, and we can use it for understanding the microbiome of the spacecraft. So here's the first paper from sequencing, and this was just a proof of principle. We used prepared libraries on the ground, which were mouse, E. coli, and lambdaphage. We wanted a nice mix of GC content. And then we also wanted to put these things together and see if we could deconvolute the 3 different organisms. So we set kind of a high bar for the experimental analysis, but it worked out very well. We were able to reassemble the entire E. coli genome just from the sequence data on board, so we had very nice coverage. We have then pushed forward with how we're going to use this, and so microbes, this is really a great place to use our sequencing technology. We can either use whole genome sequencing or we can use 16S ribosomal sequencing. So the first part is taking all of these swabs on the space station, and this is a lot more fun because you get to hang on the ceiling than any kind of swab experiment on Earth. We did a proof of principle to say, hey, instead of preparing these libraries on the ground, can we prepare them on orbit from culturing one of these swabs, picking the colony, doing the prep right there, and then sequencing. We sent these plates home as well and did standard clinical microbiology analysis and identified all 3 of these colonies with very good accuracy, over 90%. To the right microorganism. So then this was probably a little bit my fault. That was an experiment in about 2017, and I said, we're doing 3. Let's go like way higher in terms of let's multiplex all of this. Let's do 1,000 samples. And I think I kind of threw that out casually. Somebody took me seriously, and next I knew they had flown up 1,000 tubes. And I got assigned the job of we took over 800 samples all around the space station. We've got 200 left to do some temporal analysis. But this was this very high density spatial analysis of every single module and of different surfaces on these modules. We're collecting these both for the genomic analysis as well as proteomic analysis. It was a dual swab tube collection. Again, the experiments still being processed on the ground, and hopefully results will be forthcoming soon. We've got a lot of different systems to grow cells in space, and so this was another experiment that was looking at multiple different cell types, and this was culturing bone and synovial fluid and cartilage together. I mentioned the nematode experiment. This was super cool because we grew them for 7 generations. And so we could actually look at heritable changes. You can see the function here is being tested. You can see deflection of the posts. And this was wild type and dystrophin mutated nematodes compared to ground controls. And then lastly, the plants. This is a really great area to do structural studies, functional studies, and then genomic analysis. The astronauts love it because we usually get a very small, maybe 10% of the crop to eat. And this is about the most exciting thing you can have in space, is to have a radish or a piece of lettuce. So we fully support any and all plant experiments coming our way in the future. With that, I'd like to acknowledge the huge research teams. I presented some of the cardinal heart data. This was Joseph Wu at Stanford. And then the team effort led out of NASA JSC for the biomolecule sequencer, helped a lot by Charles Chu at UCSF. And with that, I will wrap up, and we can moderate questions from Lavorka.
Thank you so much, Kate. That was an awesome presentation. We have quite a few questions for you here. I will do my best to try and get through them in the time that we have. But first question here is, from your perspective, are there areas of space life science research that are underserved that would increase the safety or quality of life for crews?
Yeah, so underserved for quality of life for crews, I think nutrition is a really tough one. We are doing more and more nutrition experiments, but trying to really actively use plants as a supplement for our food on board. Right now, we've been doing plant studies to understand fundamentals about plant growth in microgravity. I think this is really important, but I would like to see it transition to things like, operationally, can we grow crops in space? How much can we grow? How much energy is that taking? How well do they do? Are there disease issues? Our crew really loved growing plants in space. We flew up some of our own seeds, and actually it had to go through NASA safety, but we had our own gardens up there in addition to the official crops in space. And so one thing I think would be great, I would hope the space station program would support, we'd like to have a whole rack where the crew can tend the experiments, monitor the watering, really figure out how to be cosmic farmers. I think that the crew has a lot of input on how these plants grow, and in collaboration with the ground, we could do a lot operationally there. So I think that would be a huge one.
Wonderful. Next question is, thank you so much, Dr. Rubins. Can you please comment on the major differences in cancer cell biology in microgravity versus ground? Does carcinogenesis occur faster or slower in microgravity?
We have done some cancer cell experiments where we're just looking at cultured cells. This has been not a huge area of study. I think that certainly we do see some properties of these cancer cells altered, but some of these were a little bit earlier experiments, and so I'd like to see more replicate studies that are looking at growth and differentiation. What we really haven't done yet is some intense studies where we're looking at tissue architecture. And I think a cancer, an experiment where we could look at tumor microenvironment, maybe vascularization. These are pretty complex experiments to grow that many different types of cells, but either on a tissue chip or on a printed architecture are the kinds of things I think we need to do. We have not delved into this a huge amount. We've really started to get into stem cell, and we can see that we get a lot more stemness, and we get much higher rates of stem cell growth in microgravity. And so I think this was a very interesting question for cancer, but we haven't studied it a whole lot on board.
All right, that's awesome. All right, here's an interesting question. If you could choose one plant and one animal to bring into space, what would they be?
For experimentation, I think the rodent models are really, they're really valuable. They're really beneficial. We always say in immunology, mice are not people, but we can learn a lot from the rodent models. So I think this is probably one of the best model systems and right-sized for a spacecraft. I think for plants, you know, the radishes were a really great experiment. If I'm really picking for biology, of course, you have to go with Arabidopsis. If you're walking the line between just a pure model system and something that you want to eat, any kind of leafy green or a radish is going to give you lots of interesting work for morphology, lots of sample production, and then is also a great nutritional source for the crew. Funny story, we get our harvest and we each get like a piece of lettuce, and we put a little drop of olive oil and a little drop of balsamic on it and made a salad, and it was the best salad I have ever had in my life because we had had no green things for the previous 4 months. So it's delightful.
Must have tasted better than chocolate.
Great.
Okay, so we have a couple more minutes here. Maybe we can squeeze in 1 or 2 more questions. Okay, so the next question is, NASA selects astronaut candidates on several different tracks. ESA currently does not. Was there any gap in astronaut training you had to overcome as a life scientist as compared to pilots or engineers? And were there any advantages?
That's a great question. So we bring everybody in from very different backgrounds. They all have some kind of, now they need to have an advanced degree, at least a master's, and everybody is, even the pilots will have an engineering degree or a science degree of some kind. But you're talking about putting life scientists with, you know, aerospace engineers and military fighter pilots. So we train the pilots to be scientists and we train the scientists to be pilots. And we really are all doing the same job on board. For me, it was definitely a learning experience to go into high performance aviation flying. I went to flight school with the Navy. I ended up getting the opportunity to go back again and to solo and to qualify as a front seater in the T6. I fly backseat in the T38, which is a supersonic capable fighter jet trainer. And so that was definitely nothing that I had ever done as a research scientist. But the pilots get a chance. We put together a course for them to do bio basics, we call it. So they get to go into the lab for a week. We teach them microbial techniques, we teach them sterile techniques and sequencing, and they really love this. So it's kind of this great intersection. And after a few years, you're still coming from different fields, but everybody is speaking this common language of training for spaceflight.
Wonderful. And maybe one more question here. Okay. So we are currently working on a new science chamber with automated media change and a microscope recording real-time data in a very small setting. What feature besides real-time monitoring and automated media change and fixation is the most important one in your experience?
Yeah, great question. I love it. I've been agitating for this for a while. I think it's very important to be able to take aliquots of the media and of the cells if possible, and be able to pull off time courses from these. I'd like to see, I'm going to give you a few things, not just one. I'd really like to see long duration experiments available. And I want to make sure that something can be used for either mammalian or yeast or bacteria. I mean, we need a chemostat on board. I really want to grow yeast for multiple generations and look at heritable changes due to spaceflight. Our radiation environment isn't quite enough to provoke massive changes during a 30-day time course, but if you're growing something for a year, you might see some really cool and interesting results. I think it's also important, in addition to the real-time light microscopy monitoring, to be able to fix these cells, stain them, and use immunofluorescence, because that's where we really get at a lot of the structural studies. We can look at architecture. If it's a multiple tissue, we can look at the cell morphology. I think cytoskeleton is really understudied on board. So I love the system. Sorry, I have way too many ideas of what we could do with it, but I think the basic premise that you've got of the automated media changes and the real-time monitoring is absolutely the setup we need to start with.
Well, thank you so much, Dr. Rubins. We are out of time. I really, truly appreciate your time. It was an honor to have you here.
Yeah, absolutely. So let me get out of this and give you back.
Awesome. Well, thank you. And continuing on to our next category, we have the GeneLab grant recipients. So this section will showcase the research proposals that were selected for the GeneLab Analytical Investigations Grant. This grant is focused on investigations that will utilize the GeneLab data system to answer systems biology-related questions regarding how living organisms respond to the spaceflight environment. There's 3 speakers in this category, and each will be followed by a 4-minute Q&A at the end of each speaker. So with that, today we will, the first speaker in this category will show a pre-recorded talk from Dr. Sushma Naithani from Oregon State University. And her talk is titled Plant Reactome, a knowledge base of plant gene regulatory networks and pathways and its use for spaceflight biologists. So go ahead and take it away.
Hello, greetings to you all. First of all, I'd like to thank organizers of this meeting for inviting me. I take this opportunity to tell you a little bit about the plant reactome database and a little bit about the recently funded project from NASA about biocuration of some of the gravity-related pathways. So we got a small funding from NASA for in silico modeling of gene networks, pathways, and mechanisms associated with plant gravitropism. We will use plant reactome platform for biocuration of the pathways that are associated with plant gravitropism, response to altered gravity, and response to irradiance. As you can see that we already have pathways biocurated in the plant reactome that are related to various cellular processes, circadian rhythms, growth and developmental pathways and processes, plant metabolism, and plants' response to various abiotic and biotic stimuli. These pathways will be placed under the plant's response to abiotic stimuli. We hope that we will be able to integrate these pathways with the overall plant pathway network that represents plant developmental pathways, differentiation of various organs, plant metabolic pathways, and plant gene regulatory pathways. By doing so, we hope that we help researchers in developing deeper understanding of how plants respond to gravity. Nonetheless, researchers who are interested in analysis of the omic data via transcriptome, proteome, proteomic, interactome, or metabolome data, they can use plant reactome as a resource for analyzing the differentially expressed genes in the context of plant pathways. Plant reactome is a pathway knowledge base or portal of the Grameen project. It is really accessible. It is an open source project and it is available to all free of cost. Currently, we have plant pathways for about 120 species represented in plant reactome database. Again, users who are working in various species can access plant reactome at plantreactome.grameen.org. So what we do here, we manually curate reference pathways using evidence from scientific literature. And for curation purpose, we use rice as a reference species. Then, using the automatic protocols based on gene orthology, we generate pathway projections for other species. So currently, we have species that range from unicellular prokaryotes, photoautotrophic prokaryotes, to lower plants to hybrid plants, representing both monocots and dicots. So thus, we represent a wide range of taxonomic clades for which pathway information is available. They include both model plants and crop plants and plants of the other interest. Here is just a view of how these projected pathways relate to the curated pathways. So as you can see, On the x-axis, we have the various species listed. Here are roughly dicots followed by monocots. Then we have the lower plants, and then we have photoautotrophic bacteria. As expected, we see less number of genes associated per reactions or the pathways, represented by blue color. There are also some gaps as well. And here on the polyploid species, we see a very high number of genes associated per reaction and per pathways. We use plant reactome data model to represent various different types of reactions that represent metabolism, signaling, transport, gene regulation, plant growth, development, and differentiation of various plant organs, and how plants respond to abiotic and biotic stress conditions. So input molecules can be any of the molecules listed here: protein, macromolecule, RNA, even DNA. And then output reactions can be any of these. And sometimes the same molecule can be input and output, but it can be transported to a different cellular location. So simply you can go to Plant Reactome homepage, and if you are interested, you can click on the pathway browser and you can access the pathways that are available in Plant Reactome portal. We use the Firefox API to organize and list the pathways. You have also choice of searching for the pathway, a metabolite, a protein, or a gene in the Plant Reactome homepage. And again, you can get that way also to the, for example, here the salicylic acid. You can get into the salicylic acid signaling or biosynthesis pathway. You can get the summary of it, and then you can get the hierarchical listing and go to the salicylic acid signaling pathway again here. So by search or by browsing, you can end up finding a pathway in the pathway browser. And as you can see that we use really a eukaryotic plant cell architecture, conceptual architecture of plant cell to depict the pathways and the reactions in Plant Reactome. And you can see that different locations of the gene products here. So representing the various subcellular compartments. Representing various subcellular compartments. By default, we have Horizon Sativa, but using this drop-down menu, you can choose a species of your interest and see how the projected pathway looks like. We have another tool in the analysis tool. If you click, you will find the species comparison tool. And here you can compare the reference pathways with the projected pathways. This comparison is for rice and maize. And as you can see the green box, which means that we find rice matching orthologs in maize, and number of the genes in maize and rice for that particular gene group or gene family are same. When you see half blue and half green, which means that you find more or less number of the orthologs in maize as compared to rice. When you find nothing, then you see the blue box. In addition to pathways, one can look into the bottom panel here on the right hand side, and you can find the pathway summary. You can also find the gene expression information of the various genes that are listed in the pathway. And this information for the basal gene expression is coming from the Plant Gene Atlas of ABI, so it is automatically fetched into the Plant Reactome database. Another information can come from the various sources of the curated gene-gene interaction data. And for example, here I have provided an example of the Arabidopsis pathway and the interaction data that is available for Arabidopsis. And as you can see here, actually you can expand this view, and here we have used this source of Plant Interactome, but then there are other sources including the bar and inect and bind and so on. So you can select and you can check more than one databases that provide gene-gene interaction data. This feature is really good because we know that manual curation is slow, and providing the overlay of the gene interaction data can help our users to formulate a much better data-driven hypothesis for their experimental research. The other feature that is useful for doing omic data analysis is that users can upload data of their interest, that is transcriptome data, metabolome, or proteomic data, and see what is happening in different pathways, and download that result as well, and find out the upregulation and downregulation of the various pathways, genes, or the metabolites. As I have mentioned that previously, that higher plants have multiple copies of the genes, large gene families, and sometimes it becomes very difficult to find the right candidate genes. Nonetheless, you can actually click on any of the gene. For example, here I have clicked on this blue gene, and here there are 4 genes that are mapped to this particular reaction. As you can see that they are all differentially regulated. Only one gene is highlighted in red, is actually upregulated. So this feature can really help you to identify the candidate gene out of the multiple gene copies and gene paralogs. So if you have a list of differentially expressed genes, using plant reactome resource, you can find their updated functional annotations, what protein products they encode, where the gene product is located within the cell, and then summary of genes and pathways that we update based on literature references, which are the associated reactions and pathways. And then also you have ability to overlay gene interaction data, gene expression data, and further learn about the genes and the proteins that are associated with a particular response or a treatment. And we provide all that within the conceptual framework of a plant cell. So we hope that by next year, we will be able to curate and integrate some of the pathways related to gravitropism and connect those to the existing plant developmental, metabolic, and gene regulatory pathways for better understanding of plants' response to gravity. And the researchers working with the GeneLab data can take advantage of that. I like to thank especially a lot of our undergraduate students at Oregon State University who have been involved in past 3 years under various programs, including honors thesis to research credit courses to the research internship that were supported by URSA Engage program at Oregon State University. So they have done a lot of good work about gene and pathway biocuration. We actually formed a virtual classroom during the COVID-19 shutdown, and it was a great experience working with all these young people and scholars. I like to also thank our collaborators at Oregon State University, Cold Spring Harbor, that is the 2 partners of the Grammy project, and other collaborators at NYU Medical Center, OICR, EMBL EBI. And I like to thank USDA, NSF, OSU, and the Plant Reactome project for supporting a plant reactome database. And more recently, I acknowledge the funding that we received from NASA GeneLab. Thank you very much. I will be able to answer your questions in the chat.
Thank you so much, Dr. Naithani, for that. Would you mind turning on your video and audio for Q&A?
Yeah, hello.
All right, we have some questions for you here. First one is there anything particularly exciting to you about the recent study that used lunar regolith to grow plants? Would you like me? Can you hear me okay?
Yes, please. So this is the question number. I can see actually Q&A. Which question is that?
Okay, it's towards the bottom. Is there anything particularly exciting to you about the recent study that used lunar regolith to grow plants?
Sorry, actually, I don't know about that study. I have to see that.
No worries. So you had mentioned, here's the next question. You had mentioned manual curation. Is that done with your internal team through the larger scientific community?
We have our biocurators who are trained and dedicated to the projects. We also have undergraduate students, and we have also some of the community curators, yes. And we are happy if anybody like to curate or learn how to curate pathways, or if they like to review the curated pathways. So we are open to the community biocuration.
Wonderful. Here's some more questions here. Were there any studies about changes in chromatin condensation in space, and how might that affect expression?
I am actually not aware of any such studies that deal with chromatin condensation. So last time when I checked the NASA GeneLab data, I did not see any such data.
Okay. What is your favorite visualization on the plant reactome?
I think the omic data analysis is pretty good, and I really feel that the ability to see how paralogous or orthologous genes behave in terms of their expression in response to various treatments or during the developmental stage, that is my favorite feature of plant reactome.
Awesome. We have one more question here. Is there a way to integrate the tissue-specific nature of the gravitropism response using the reactome pathway browser?
Yes, there is a way to do that, and we could actually have the parallel pathways. Some of the examples that we have done in terms of the tissue-specific response is related to the other abiotic stress, for example, submergence. So we are doing the submergence response, for example, in roots, in shoots, and what happens in the young leaves or germinating seeds and so on. So we can have those, and yes.
Wonderful. Thank you so much. If there are any further questions, oh, here's one more question that came in. We probably have time for just this one, and then we have to move on to the next speaker. Are there any graduate student volunteer opportunities to participate in your group via the AWS network?
Yes, sure. So if anybody like to volunteer or collaborate, you are welcome.
All right, wonderful. Thank you so much, Dr. Nayathani. We really appreciate your time. I know it's late in India. Again, many thanks.
Yeah, thank you so much.
All right, so our next speaker is Dr. Sarah Wyatt from Ohio University, and her presentation is titled "At the Intersection: Meta-Analysis of Transcriptomic Datasets." Welcome, Dr. Wyatt, and take it away.
Thank you. Can you see the title slide?
Yes.
Yes, we can.
Which obviously has a different title. So we'll get to this one as well. So this is a prequel to the GeneLab analysis proposal that we did at the intersection. So this was an experiment that we did several years ago. Colin Cruz started it. He's no longer in the lab, obviously. He's at LANL now. But the idea of taking 3 data sets, part of them from GeneLab, using GeneLab data to look at an intersection of genes expressed in gravitropic responses. So the first one that we chose, this is a ground-based microarray. This is system that I developed years ago, the gravity persistence signal. And it's simply a way of looking at signal transduction. So this is on the ground. We did a time course over the 2, 4, 10, and 20-minute time period, which should be the early signaling events. Again, that was a microarray experiment. The second experiment was a spaceflight transcriptome. This is brick 20, the first spaceflight that my lab did. These were done, as you can see in the upper left, or kind of mid to upper left. These were bulk planted Arabidopsis seedlings, and they were in the brick hardware. So these had no light, and this is what we returned. So these were frozen on flight. And what you see in the lower right are the little seedlings or etiolated seedlings. Again, the work done by Chromatin and Colin Cruz. The third data set that we used for this was a space flight transcriptome, another microarray. So we have 2 microarray studies. We have one RNA-seq, and this is a research project. This is GLDS 7. So these were the early days before there were a lot of data sets on. And this was by Rob Furl and Annalisa Paul. So these were 12-day-old seedlings done in the apex tags. So these had light. They're a little bit older than the brick 20 seedlings, which were 3 days, and again, etiolated. So really, environmental conditions here are totally different across all of them. The only thing that is consistent is that there's an altered response to gravity. We have 2 that are microgravity and 1 that's a reorientation experiment. So what we did, just to recap all this, you've got 2 microarrays, you've got 1 RNA-seq, you've got 2 space flights, you've got 1 ground control. And what we did was analyze all of these and look at what genes were expressed in multiple, in all of these sets, with the idea that those genes must be fundamental to gravitropic response. And what we wound up with, believe it or not, were 5 genes. 5 genes that were either upregulated or downregulated, differentially expressed in all of the data sets. If they were, interestingly enough, if they were upregulated in space, they were downregulated in the ground. So they had opposing expression depending on where they were. What you see here are 3 proteins involved in ubiquination. So you have the F-box protein at the top and the 2 E3 ligases. And then the 2 in the center are both transcription factors. Both of these, obviously, all of these in control of gene regulation. And just to try to be sure that these actually have an effect, we started with just a simple mutant analysis. We ordered Salk insertion lines for each of the genes. These are the 2 transcription factors that I'm showing you. And you can see that wild type is in black and the mutants are open circles. And what you can see is there is a differential expression here. So from that analysis, we're continuing work on additional mutant analysis. But for the transcription factors, we're doing ChIP-seq. And for the E3 ligases, we're looking at protein-protein binding, substrates, and hopefully that work will be completed in not too long. From this, we also wanted to look at other meta-analysis of multiple omics data sets. And this is what led to the proposal to GeneLab last year. So the comprehensive meta-analysis. This was a proposal designed by myself and Al Myers, who was a postdoc in my lab at the time. He's now at Kennedy Space Center. The basis of this, as you can see on the left, were 5 different data sets. PGP spaceflight that was conducted by Chris Wolverton and contained both wild type and the PGM mutant, the starchless mutant. So this was plant gravity perception. So you see the top one, we did the genomics analysis for this, the PGP spaceflight. Then there was a PGP Aztec that we put together. This was at the facility in Norway, looking at ground simulators of microgravity, if you will. And we'll get to a little more into that in a minute. And then the 3 seedling growth experiments. These were done by Haraz, Medina, Kiss, and Vanderbrink. So these are 3 growth experiments that are parallel. These were all done in the EMCS hardware, which we'll see in a minute. And they were done really to look at light expression. So how light responds. So let's take a look at the 2 hardware involved. You have the EMCS, which for those of you who know has been decommissioned now. So these are some of the kind of using data sets. So this is one of the benefits of GeneLab. We can use data sets from experiments and from hardware that no longer exist. So as you can see, the idea of EMCS is it is a centrifuge basically on flight. So you're in 0G or microgravity, and then you can spin this at multiple different levels to generate different simulated G forces. These seedlings are in small cassettes. You can see 5 cassettes per arm of the machine. In addition, we use the clinostats at Aztec in Norway to give us simulated microgravity. So who wouldn't want to go to Norway to do a set of experiments? These were set up. This is a random positioning clinostat. So you can see it's spinning on 2 axes. So this gives you sort of the background of these. If we look at the different experiments, so each one of these boxes contains the experimental growth parameters. You have seedling growth 1. I'm going to start at the bottom here with the orange colors. These are the seedling growth experiments. Again, all on EMCS, on flight. So you have 2 that are blue light treated, 1 that's red light treated. These were processed as whole seedlings. For seedling growth 1, you had 4 G levels. That gave us 4 distinct transcriptomes. For seedling growth 2, there were 3 G levels with 6 distinct transcriptomes. And then you have seedling growth 3, which again, whole seedlings, but 4 G levels, plus 2 mutants, Phi A and Phi B, which are light mutants, which resulted in 8 distinct transcriptomes. If you look at the 2 blue boxes at the top, the one on the left is PGP spaceflight. This again on the EMCS. So all of those were in the same hardware. You have 2 genotypes with the PGP experiments. You have Columbia, which is the background for the PGM mutant. PGM again, phosphoglucomutase, pretty much eliminates the starch in Statilis. There were 4 tissues here: root tip, mature root, hypocotyl and cotyledon. So it was dissected into all 4 tissues. These were 4 days at 1 G and a couple of hours at the partial G in the dark. There are 12 G levels here, so 96 distinct transcriptomes by the time you look at the gene level, the genotype, the tissue, and the G level. For Aztec, we wanted to use the same setup that had been used for the PGP spaceflight, so it would be as close as possible. These are simulated fractional G on the ground. There are 2 genotypes again, Columbia and PGM. 2 tissue types. We just did root tip and hypocotyl because those are the 2 most responsive tissues. So these were rotated in 2 different directions, so a vertical and a reoriented. It depends on how you place them on the machine, which way they're rotated. So there were 4 simulated G levels. This gave us 40 distinct transcriptomes. PGP Aztec Those data are not currently on GeneLab, but will be shortly. We had trouble getting those back. If you might imagine, over COVID, I don't know how many of you had problems with facilities and return of samples, but we certainly did. So what were the aims? First, to look at identify core trends in the transcriptomic response to a partial G. So the question, are similar trends observed across these experiments? So looking at PGP flight, Aztec, SG1, 2, and 3, do we see very similar trends? Again, kind of taking all of those other parameters and using them in some way as a filter. So we can filter out light because we have those that were in different light regimes, and looking at the microgravity versus the simulated microgravity. Second was trying to leverage the diversities of these experimental treatments, again, to delineate some of the influence of the other features. So on the first one, we're looking to use that diversity to really focus on gravity and what might be there that is specifically gravity. And in this aim, we're trying to look at those that are potentially other influencers. So are there G-level thresholds across gravity sensing, no matter what the other factors are? What are the impacts of the various mutations? So again, we have 2 light mutants, we have 1 starch mutant, and we have various different light treatments. So can we look at those impacts and how they impact those gravity effects? And then what are the ecotype specific impacts? So the difference between Columbia and Landsberg erecta. So for this one, we're using just 3 of the data sets, PGP flight, SG2, and SG3. And then finally, for the third aim, we're trying to use the series of ground controls for all of these data sets to understand better the utility of the simulated partial G studies. So how does simulated partial G compare to this partial G of spaceflight? And can we observe the same trends across these different G levels in the ground analogs? What are the space-specific effects? And how do those change with G level? And then finally, can we identify a molecular mechanism for the mitigation of some of these space stressors? Again, this one we're using 3 of the data sets, PGP flight, PGP's Aztec, and the SG3. So the analysis plans, differential expression, straight up, contrast analysis, impulse DE2, and MA-SIG-Pro, which those 2 both are used predominantly for time course. We're going to use them and substitute gravity level for time to look at expression across the gravity levels. And then of course, a WGCNA. And with that, I will end with my acknowledgements. So the Wyatt Lab again. So Emma Kennedy is taking over analysis of this project. Samantha Farouche is looking at the F-box proteins. Calvin Coffin is the one doing the ChIP-seq work. And Lanre, Victoria, and Wafa are looking at other aspects. My collaborators, North Carolina State and Texas A&M, and of course, all the people at Kennedy that help us with all of these spaceflight experiments. And thank you very much.
Thank you so much, Dr. Wyatt, for an awesome presentation. We do have some questions for you. The first one is in your experience, what are the limitations of clinostats or other sims that force you to instead run the experiment on the ISS?
So the reality with the clinostat is, we talk about it being simulated microgravity. but there's not been clear experiments done to see how much it simulates. You know, when I talk to children about this and do outreach, I look at the clinostat and I say, you know, this is really interesting. And granted, the plants don't know which way gravity is, but it's more like they're dizzy. So there is gravity, and it's like rolling down a hill. You're dizzy, you know gravity still exists, but you're just not sure where it is. So we're really interested to use the experiment that we've designed here to actually test what gene expression looks like in the two, and if there are really similar, or what caveats we need to have when we do clinostat experiments.
Awesome. Maybe we have time for a couple more questions here. Have you looked at the cytoskeleton dynamics in the IQD21 during gravitropic reorientation, microgravity, and/or hypergravity?
There's a really simple answer. We have not. That's a great suggestion, but we have not done that.
Okay. Another question here is, were there any genetic responses observed in the RPM that were also found in the true microgravity of spaceflight?
Can you say the first half of that again? Question?
Were there any genetic responses observed in the RPM that were also found in the true microgravity of spaceflight?
Okay, so this is sort of when I told you that the ESTEC data were, quote, behind. We just received those like a month ago. We finally got all of those data back. So we've not actually done those comparisons. We've done some of the initial analysis of the others, but unfortunately, until we got all the data sets back, we couldn't actually do much of what we had planned here. So we've done initial characterization, but we've not actually done that comparison yet.
All right. We've got one more question here. Are the targets of the F-box proteins known?
They are not. One of my colleagues here is a world expert on ubiquitination and F-box protein substrate, and none of them are known. And he's confirmed that. They're working on one of them, which is kind of nice because that way we can at least collaborate on that one and not have to start from square one looking for the binding partners. So that's really helpful. But no, all of these are, the substrates are not known for any of these.
Well, wonderful. Thank you so much, Dr. Wyatt. We really appreciate your time and your presentation.
Thank you.
All right, and our last speaker in this particular category is Dr. Pankaj Jaiswal from Oregon State University, and he will speak about the plant's transcriptome modulation in response to microgravity. Take it away, Dr. Jaiswal.
Thank you. Can you hear me and see my screen?
We sure can.
Wonderful. Right, just a second. Thanks everybody. Thanks Lavorka for giving me an opportunity to talk about our new project that just got funded. I'll be talking on the plant transcriptome modulation in response to microgravity. And I'm at Oregon State University. So if any of the folks that are interested in collaborating or sharing their data or would love to work with us, I would very much look forward to working with you all who are interested. So here's the project that is, the actual title of the project is Alternative Splicing and Transcriptome Modulation under Spaceflight Response in Plants, where we want to see that beyond the gene expression data, can we reveal some more intricate novel findings from the transcriptome data, and can we use it to understand the effects on gene structure and function in response to microgravity. Since this study is basically, this project is basically meant for studying the existing studies in the GeneLab, so we will not be carrying out any new space biology data, but it will be working mainly on the existing GeneLab studies that are present, that we have. So in GeneLab studies, majority of the times, or in any of your experimental gene expression data sets that you're looking at, often the story concludes with the differential gene expression analyses and gene ontology-based functional and pathway enrichment data sets, and then to go along with it. But we wanted to see, can we explore these things a little bit more? So we said about we will try to develop an alternatively spliced gene atlas for the plant data sets. And if at all possible, since a part of that experiment was conducted on in the microgravity environment in space, so we wanted to see if there are any cosmic radiations that may have created some mutations in these genomes, and see if we can pick it up through these existing data sets that we have. So the team for the project is myself, Parul Gupta is she's a postdoc in the lab, and Justin Elser is also a postdoc, and both of them work on the bioinformatics aspect as well, representing some of their work. The collaborations are with the GeneLab group, with John and Richard Barker. You will hear from him later on, and as well as from Colin. Both of them lead the plant AWG group, and we are also part of that group. And I'll tell you a little bit more about what do we learn and how do we synthesize this project before we came across these things. The data that we will be generating will be going into the NASA GeneLab and some other publicly available resources like BAR for Arabidopsis, ensemble plants at EMBL EBI UK, and Gramine database at Cold Spring Harbor Lab that we are talking about, kind of things. So it will be a truly public resource that we will try to generate. And the broader impact would be is some of the methods and workflows that we will develop can be applied to non-plant models as well. So a little bit about how this project came along is that 2 years back, we started working with the NASA GeneLab plant group, and we looked at the time when some of the new data sets that were already available in the GeneLab, they have been analyzing. So the conversation started happening along with that thing part, and I'll tell you a little bit more. But the group does is they identify new data sets and analytics, and Sarah is also part of that, we just heard, and asking over the new biological questions. Partners discuss a lot of future experiments, planning and updates, as well as devising strategies on how to analyze the data sets in different dimensions that we are looking at it. So it's the work. The group is open to invite new members and encourage all the younger faculty and the young investigators to come and learn more about the space, plant biology in space, and help us coordinate. So they are also allowed to mentor and do lead some individual projects that they are interested in. So the meetings happen, you will hear more later, but the meetings happen every other Wednesday between 8 a.m. or 12 p.m. There are two different meeting times that we're looking at. There's collaborations are more than 60-70 people on the plant AWG group, and we all discuss and come together in these meetings to devise, explore the data sets and go along, find something new. So as we were discussing, when we picked it up, when we came into the group was doing the microarray and the RNA-seq comparative studies on the ISS and microgravity treated samples. And you can see that there is a nice platform that has been developed now and available through the GeneLab is to look at the gene expression data sets and the functional and the co-expression networks and the functional enrichments of those kinds of things. But as they're looking at it, some of the pictures were shared by Richard Barker. And you can see that some of the prime suspects over here, that with the good quality data sets with the enough number of replicates, you can see that there were differences between the hardware that we are looking at it and obviously the type of seeds and that went into this. So it's a little bit hard to compare the data sets. Within the study, it's absolutely possible to study all the molecular events at the transcriptome levels that we're looking at it. But when you go and compare across the missions, across different experiments, it's a little bit hard because they came from different, they were grown in different hardware, different growth stages, different environment and things like that. So we would love to have some more consistency and more long-term time series experiments. These are majority one single time point studies that we have been done in the case of plants. So that's something which we will be looking at it. And as they compared, the group compared the analyses of the data coming from microarray and RNA-seq in Arabidopsis. So you can see that these two methodologies, sequencing and expression, the methodologies are relatively separated out into the two different type of experiments. And you do see a lot of these stress resistant and stress tolerant responsive gene enrichments that in these two experiments happen separately. But as we were looking at it, the group, as a group, we thought about, let's focus on RNA-seq because you can explore a little bit more about that data sets and go deeper into what else is happening in the transcriptome. So we thought about devising a project on transcriptome modulation, looking at not just the protein coding, but also looking at non-coding and small RNA aspects. And we were also suspecting that majority of the analysis currently happens on what is present in the reference genomes, but not necessarily on finding novel transcribed regions in the genome, which may be seen under the ground control or under the space microgravity environment, which we may not have seen it earlier. Some of the other studies may be coming along, but since this project is mainly funded for looking at the existing GeneLab studies, so we try to focus on the top 4 or 5 aspects that we will be looking at. Okay, so here's the first aim was, can we look at the alternative splicing of the and the transcriptome modulation? So we already have the studies available in the GeneLab platform. You can pull together either the individual sequencing read files or the BAM alignments and the differential gene expression data sets. Those are already done in there. So you can pick up from there some of the BAM alignments, for example, to carry out some of these alternative splicing analyses on our end. In that way. So you can see that there could be a pre-mRNA which is spliced properly, and then you can be skipping aspects of the exons might happen over here, or you might even see an alternative 5' splicing site. So you would have a little bit more or longer extra part of the intron, or this exon 1 could be extended kind of things. Or you might see some of the additional 3' ends would be coming into that. So there are alternative forms of these transcripts that can come. So whether they get translated into a different open reading frame to create a new protein, or they could be just an adaptive feature that it is just waiting until the stress adaptive feature that it will splice as soon as the conditions favor in that sense. So these are different mechanisms that the plants do carry out. So about 45% of the genes in plants, 45 to 60% of the genes in plants do undergo this alternative splicing, compared to humans where about 90% of those things are 90% of the genes undergo splicing kind of thing. So this is one way of creating adaptation and stress resilience into the plant, because the core set of genes is the same. This is how you create the diversity of transcripts and the downstream coding regions and the downstream proteins, for example. So here's a quick look at the GLDS. If I say GLDS, means it's a GeneLab study, number 38 and 37. Both of them had one of this particular data set that you're looking at is from the same ecotype Columbia 0. And you can see that there are more intron retention events on chromosomes, since you're looking at it in general, compared to the ground control, which is only this thing. So there is a heavy amount of, there is a little bit more number of alternative splicing aspects you can be seeing in the flight data sets that in general we're looking at it. 37 turned out to be a little bit the similar way that we are looking at. Both of them, these experiments, one of them I know that it's a 3-day seedling growth stage, and another one is an 8-day growth stage, and both of them I guess have the same, have a different mission, obviously different missions that we went in. 37 was done along with 3 additional ecotypes. So we have a little bit more comparison across the diversity of ecotypes, or diversity of different varieties of Arabidopsis that we are looking at in general. So here's a quick look of the 38 study that we were looking at it. You can plot, this is just a chromosome view of the chromosome number 1. You can see that there are about 214 splice genes that are also differentially expressed, and these are the interesting ones. These data sets, you would not be able to pick it up in only if you're looking at the differential expression aspects. 587 of those flight data sets, and were compared to 530 in the ground control of these gene sets. And by extending this analysis, we were able to even look at the splicing data sets in the non-coding RNA aspects. And so we find somewhat close to around 20 to 16 linked RNA data sets that were unique to ground control versus flight. And then 3 of them microRNAs and some tRNAs were also undergoing splicing. So these are the interesting things that we will start looking at in detail as we go and explore many of these data sets. Okay. So just a comparison of 38 versus 37, you can see that there is a core set of about 8,000 genes that are fairly conserved in their splicing aspects. But there, for example, these 646 ones, they are mainly, you can see that they are present in 37 and the 37 and 38 flight that are unique to those things. And you can see many of them are showing up for the cell wall aspects because cell wall is one of those things that provide more stability and to the plants in general under gravity that we're looking at it and strengths. But in the core set, you would see the auxin transporters and some of the oxidative stress pathway genes that are coming out, these kind of things. These are auxin ones are pretty much involved in the development and the gravity response for both shoot and the root development. So here's, I'll show you a quick view of the Genome Browser where we're trying to put together all the BAM alignments. Sorry. Oops. We have, you can see that over here, there's one gene, SCAR1, which is actually involved in a complex that is involved in actin enucleation aspects. You can see that into the ground control, under the flight, there is a shorter representation of coverage for the 5', sorry, this is the 3' end of the gene, and the ground control has a little bit longer 3' end that we are looking at. But over here, if you look at it, there is transcript splicing differences. If you see the dark ones, these are splice transcripts. These transcripts splicing events were not represented in the reference genome. So these are the new and novel events that have been discovered in this data sets. And then there could be exon skipping aspects that we are looking at it. So the whole of this data sets would be shared with the GeneLab and the community through the Genome Browser, and you should be able to upload your own data sets or view, switch on and off your own data sets or any of the data sets that we are trying to be pulling it off from there kind of things. So the aim number 2 is on the SNPs. We are still trying to figure out which is the best workflow to look for those kinds of things, because these are not the typical SNPs that you would be looking at, because there could be some other events that may be able to figure it out. But the first try at the GATK pipeline that we've been using is to get, instead of doing, we have been using it for SNP identification. So we don't have to do much of the first part because GeneLab has already done much of the interesting work of the genome alignments and regional alignments that we're looking at it. So we just take those alignments and pass it through the rest of the steps of this pathway and the workflow. And we do identify the SNPs. Once you have the SNPs, then we can always go to Gramine and ensemble plants from EMBL, EBI, and Cold Spring Harbor to look for the variant effect predictions on the existing genome annotations that we are looking at it, right? So here's a quick look at the 38 flight and 38 ground control things that we're looking at, just the coding regions only. About 55% of the SNPs that we identify are synonymous over here, and about 44 to 46% of those are missense variants. And these would be interesting ones if we taking a look at it. So taking a look at on the Genome Browser, you can actually see when you plot these alignments that wherever there you have a consistent SNPs between the ground control and the flight data sets, that means your original samples, the seeds that were sent in, were basically more or less the same samples, and there is no problem in that sense. But if you take a look at it closely over here, There are a few more SNPs that are gained in, and those are the new SNPs, potentially are the new SNPs that have been gained in those flight samples that we are looking at. So we will be exploring more intently on those kinds of things. So here's a quick view of those, because you would see that some SNPs have been showing up in the flight. We have seen about anywhere from 1 to 10% of the reads do carry some of those SNPs that are maybe unique to the experimental setup and not necessarily were present before the experiment was done. Another one would, you will see that there are some additional ones which are splicing aspects that will be happening. So that's principally is the last slide, and I'll be happy to take the questions from you onwards. Thanks.
Thank you so much, Dr. Pankaj. Perfect timing. All right, so we have a question here for you. So my interpretation of these results suggests to me that we need to agree upon a standardized hardware or plant model and perfect that from a biophysics perspective. Do we know how to reliably grow healthy plants in space? And do we need a series of plant science reference experiments to demonstrate viable plant production in space?
I think that's a really, really good question. And that's a question also for the folks who are funding and planning their new experiments and planning the infrastructure aspects of this ISS and new other orbitals, machines and things that will be going up on there to kind of things that we need to do a lot of standardization beforehand kind of things that we're looking at. But in addition to that, I'm thinking that what we show as a ground control study is where all the other environmental parameters are replicated into the ground control minus the gravity. That's the thing that is happening. So we think that it's the only factor. But if I would suggest that we need to have one more ground control where you have simply the regular under the ground, under the regular pressure and the gravity, but using any of the regular growth chambers, because we don't know the comparison, because higher amount of CO2, higher amount of pressure might be leading to other kind of fluid dynamics that we are looking at it, and that may be leading to some of the other things that for example, we were seeing RNA slippage or DNA slippage, DNA polymerase slippage, and they might be creating the SNPs in the transcript that we're looking at. So I think there is a lot more standardization needs to be done in that area.
So you could have already answered this next question, but this next question is, what aspect of the flight environment do you think could be causing new SNPs?
We think the original question was that we think that the cosmic radiation was the possible cause. And considering that we did not see the SNPs in all the transcripts that were coming along, right? So only few of the transcripts. So that's a drawback of the sampling that we have done, is because it's a whole shoot or whole aerial part of the plant, right? And only some of the cells may have received that cosmic radiation. So and we are macerating the whole tissue of the shoot sample to see it. So you don't see it consistently across the transcripts evaluated from all the cells and tissue types that we had. Only few cells had carried it. And so we do see some reads showing up those SNPs, but are they because of cosmic radiation, or are they because of high CO2 concentration, or is it because of the RNA slippage usually that I showed you in one of my slides? Is it because those slippages usually happens where there are a lot of repeats are happening. So wherever you have indels, that means there is a lot of slippage over there. And then there are a few more. So I think definitely there's a lot going on. So as I said, there needs to be one more control experiment, which is the regular Arabidopsis as you would grow it in any of your regular experiments that you would be doing it, not necessarily replicating what's happening on the ISS. Thanks.
Wonderful. Thank you so much, Dr. Jaiswal. We will have to move on to the next speakers. So the next category are the AWG status updates. In this section, the elected leads from each of the 5 analysis working groups will provide a 10-minute update for each of their groups. There are 5 speakers in this session, and as this is meant to be an update, there will be no Q&A. So with that, our first speaker in this category is Dr. Nate Shefchik from Ohio University, and he will be giving a status update on the animal AWG. Go ahead, Nate.
Thanks, Lavorka. See if I can manage to share. No, I'm getting different share options than we did the test before. All right, you can see the slide now?
Yes.
Yeah, sorry, it just didn't give me the same share options as before. Anyhow, thanks for inviting us to do the update. For those of you who don't know, as the name implies, the Animal Analysis Working Group basically analyzes animal data sets from GeneLab. There's a lot of different elements to the working groups, as mentioned by John. So I'm not actually going to start with the papers, but rather I'd like to start with some of the other accomplishments. So if I've got my numbers correct, in the past year, we've got 8 AWG members who've obtained their first permanent academic post, which I think is outstanding. And again, if I've got the numbers correct, I think that is 3 here in the US, 4 in Europe, and 1 in Japan, which I think highlights the fact that the AWGs are really a great platform for open science and for breaking down both cultural and geographic barriers to performing science. We've heard people talking about the idea that we need new data sets and we need to fly things. So in that respect, it's a great privilege to be able to say that 2 NASA flight grants were awarded to animal AWG members. One of these will be generating some gut microbiome data for C. elegans, and we'll also be looking at genetically diverse C. elegans and the interaction between the microbes and the C. elegans. And so the PI on that is Siva Vanapale. So for those of you who saw Kate Rubins talk, he's also the PI for that strength experiment that she was showing. And Kate, I can tell you that the worms are actually weaker in space. So do feel free to reach out to Siva if you want to chat about that. The gene expression is still ongoing for that. And again, in terms of the other AWGs, Ryan's going to talk a little bit later about the ASDLA analysis working group. And that sort of data that we've got for physiology in worms, coupled with transcriptomic data, is one of those sorts of projects that sort of group might work on. There's also been a flight proposal accepted for looking at changes in the central nervous system and muscle in Drosophila and C. elegans. And that's actually particularly exciting in the context of space exploration, because the key things that are being looked at in flight that are new are the effect of lunar and Martian gravity, and that's to just push things along. As I said, we've had some papers that are published, 4 in the kind of reporting cycle that we've got. The first one there in the International Journal of Molecular Science is a good example of a paper that has used existing GeneLab data sets to try and gain some more insight, much like the previous speakers have been talking about. The next one is actually another service that the AWG provides, which is if there are PIs in the NASA community who want to do transcriptomics and they need help doing that sort of analysis, the AWGs are happy to provide that. And so that's what's happened in that scientific reports paper there. And then one of the other things that we do as groups is we work together. So it's not just the AWGs in isolation. So we, along with the other AWGs, worked on establishing a consensus pipeline for use by GeneLab. And then again, the bottom one is another example of the AWG helping a PI who had existing data that she wanted to get processed. And again, it's nice to be able to help people and build a community. On the community side of it, you know, we are also looking to spin out new collaborations. So previously, the AWG has helped establish the European Space Agency Space-Omics topical team, which I'll point out actually has a special issue in Cell Press coming out this summer. That includes a particular policy piece on the use of omics for European Space Agency astronauts. And also the International Standards for Space Omics Processing Group, which is really establishing a platform for NASA, ESA, JAXA, and other space agencies to work on having unified approaches. The new spin out this year is we think there will be a single cell analysis working group that will be probably giving an update next year. And we've also had a couple of invited talks within the NASA community. And with that, I will turn it back.
Great. Thank you so much, Nate. I really appreciate that update. And our next speaker will be Dr. Brayden Tierney from Weill Cornell Medicine, and he will be giving us a status update for the Microbes AWG. Take it away, Brayden.
Hello, everybody. Thank you so much for the chance to talk. I really appreciate it. So yeah, my name is Brayden Tierney. I'm one of the 3 co-chairs for the NASA Microbe GeneLab AWG. The other 2 co-chairs are Danny Besden and Nitin Singh. We sort of came in, the 3 of us, and John Galazka is our NASA touch point. We came in January 22 and have had a really, really fun time sort of setting a bunch of, setting the course for the Microbes AWG for this year. And I have a bunch of fun updates to go through. First off, some data updates organized by Richard Barker. As we have a whole bunch of microbial related data sets. So it's not just bacteria, it's really just about everything across a range of different organisms, mostly relating to spaceflight, as you would expect, with a range of different measurement types, from transcriptomics to metagenomics to sequenced isolate genomes, all sorts of things in there. Here's another view of the different assays that we have on GeneLab, and this data set is continually growing in members of the team in the GeneLab Microbe Working Group, of which there are about 75 of us, are continually analyzing individual portions of these data sets. But what I'm really excited to talk about today, and this is just because this is something that gets me very excited, is the sort of initiative and process management workflow we set up for publication and for organization in 2022. And I know it's probably good you're all muted, because the words process management get people very excited, or at least they do for me. But to sort of contextualize what that means is, we really want to answer the question, how can we make the Microbes AWG the #1 group out there on Earth and beyond for space microbiology? The chairs sort of put our heads together and thought, what is the roadmap to getting to that point? And so we laid out, and this is just how I like to think about these things in kind of a funnel structure, starting at the top with 3 really big core key initiatives for the year. #1, optimizing our communication and project organizational strategy, building out an organized code base and website, which we unfortunately don't have time to talk about today, but there's a lot of progress being made there too in collaboration with other groups. And of course, optimizing our publication workflow. We then have split these into quarterly. These are just our Q1 examples, key performance indicators or KPIs, sort of making sure we have clear tasks to get through. Again, this is sort of how we're organizing our funnel. So we have the big picture, the big 3 things, then subtasks underneath each of them to really make sure we're executing on and delivering for the AWG to maximize its impact worldwide. And so, examples of stuff like, we really streamlined the membership process, made it really clear. Here's how you become a member of the AWG. All of our talks now have our little QR codes, so it's very easy to sign up. There's been some really simple stuff, implementing a really effective Slack channel for project management and organization, making sure everybody is involved, engaged, able to communicate, and feel sort of welcome to communicate on any of the ongoing projects. Then I want to talk today a little bit about how we're doing our sort of publication and organization workflow. So we really wanted to think hard, because it's not a trivial task, right? Like, we have everybody in the AWG's put out a ton of publications coming from small collaborations or individual labs, and that's really cool. But what we wanted to do was scale that to make it basically, how can all 75 people, how can everyone be involved and take part in taking an idea from idea to actual scientific product to executed publication, and how do you organize that? Because it's quite hard, but we have all these world experts, and so you'd think that we should be the #1 place to do something like this. So we really came up with clear strategies for task ownership, deliverables, and timelines, leads for each publication underneath sort of the 3 pillars of GeneLab: standards, pipelines, analysis, and reviews. our goal are to have 3 AWG microbe-wide publications completed at least by the end of this year that span these 3 different categories. And so sort of the real question, and again, I know you're getting a lot of process management. Like I said, this is something that excites me, and I think it's something our group takes a lot of pride in terms of how we've structured our organization. It comes down to how do you operationalize consortium scale scientific output? It's kind of, and we basically come up with three really clear principles, and we design the workflow I'm going to show you on the next few slides. It's clear task ownership across the entire process. So where does the buck stop? If I want to be involved in the writing the discussion section for a paper, how do I know exactly who to talk to? Well, making that really clear at the inception of a project and assigning ownership and incentivizing that person to really be excited to work. A straightforward task path between low and high resolution tasks. That's that funnel talking about. So you have the really big idea, like we're going to answer this one big question, and then zooming down deep to, okay, well, one increment, one grain of sand to go towards doing that is doing a big lit review on this one topic, and then standardizing inputs and outputs. What is a reproducible format for lit review? So this is again, I know it's sort of conceptual, but we're going to get into brass tacks in the next few slides. It really gets at that question of if you're in a larger organization, how do you systematize science in a way that it's accessible to everybody involved? Communication lines are clear, everyone knows what's going on. It's very easy in a small collaboration or lab to have things to evolve organically. But when you really think about how when it grows to something the size of the Microbes AWG, we really wanted to find a way to systematize the entire process. And so the way we sort of have worked this through is, because the chairs really serve, at the pleasure of everybody in the AWG, we want to make sure ideas were coming from them. So we set up a workflow for querying the AWG for topics, figuring out what our strengths and weaknesses are, integrating and presenting findings to the chairs, generating publication-specific subgroups, within those subgroups, assigning leadership and ownership and accountability to each of the tasks required to get a publication out. So here's an example. We queried the group, figured out what are our primary skill sets, and we were not surprised and pleased to find that it covers this whole range of different activities and different skills. We're good at a lot of different things. And then the question became, okay, how do we integrate these into creative new scientific ideas? So we started asking the group writ large, what areas do you want to work on? What really gets you excited in terms of actually generating scientific output and publishing papers? Are you interested in leading or participating? You can see there's sort of a distribution here. People really wanted to write reviews and do analyses, which sort of led to what our initial projects were. Then we started looking at different projects. We proposed a bunch and gave people space to propose others. And people were really excited about writing reviews on how microbes are going to get us to Mars, or a review on the past, present, and future of spaceflight, really thinking about these big ideas. Also, some people wanted to look at specific data sets like GLDS 249, this rodent research data set that has a ton of metagenomic and microbiome data. And then also there's some sort of more cutting edge, wilder stuff like looking at AlphaFold or phages or viral analysis. And so we, the chairs, took this feedback and then proposed a series of potential papers that could be written. And for each of those papers, we developed these subgroups, one based on review writing, that's actually should say review writing, and one for GLDS 249, one for rodent research. That's what that middle box should say. And then in fall 2022, starting a viral subgroup for the rodent research group. This is led by these 4 amazing individuals, Nick and Mike. Is that time, or 2 minutes, I think?
2 minutes.
2 minutes, lovely. And so you can see Nick's going to talk about this later. He has really done an incredible job at taking ownership of this project, and we're pretty far into the process of getting a paper written on it. It's this incredible cohort of mice that were sent to space compared to the one that were on Earth. And we went through, and this is again that funnel process where we've gone through and assigned tasks and had people take ownership over every single component of this manuscript writing paper. So now we have we have engagement from the entire group, and we have clear inputs and outputs saying, this is what a lit review looks like. This is what a data packet, this is what an analytic unit looks like. And so this is how we actually can move forward and operationalize this entire process. So in summary, there's a ton going on in the Microbes AWG. We're thinking a lot about how we can scale and sort of hit functional economies of scale within our group to be able to churn out a lot of really high impact publications this year. We have a ton of stuff ongoing in the background, also individual people working on individual things, but the AWG wide papers are what I'm really excited about. working on the systematic approach to paper writing and publishing. We're growing the code base. We really want to collaborate and grow the group. There's so much work to do. There's so much really exciting stuff to think about. So, and we also have a lot of people to thank, funding from NASA and GeneLab and the space biology program. And so please feel free to scan that QR code, fill out the form, and we'll get you added to the Slack and get going on some cool projects. Thank you very much.
Awesome. Thank you so much, Brayden. Our third speaker in this category is Dr. Afshin Beheshti from NASA Ames Research Center in the Broad Institute, and he will be providing an update on the Multi-Omics AWG.
Okay, thanks, Lavorka. I had to unmute myself first. Let me hide this and let me go. Okay, so let me jump right into it. Currently, this is our little profile. We have 180 members. Not all are constantly there, but people hop on and off when they see interesting projects. So we see this is the little map in the US and Europe and Asia and South America and Australia and so on, Africa. So, you know, hopefully we could fill in all these countries eventually. That's the goal, get a universal global output for multi-omics and how to use that for space biology research. But here's the overall goals of multi-omics, as I'm sure people already know, but you know, we try to look at all the omics and how to eventually incorporate them that together. Part of it is like, you know, see if we have pipelines. The other part of it is just analyzing data, just incorporating proteomic data that's on GeneLab, transcriptomic, epigenomic, and so on and so on. So that's the whole point. We take all this data, put it together, and then we come up with some really interesting, fun papers and projects that people come up with, and I'm going to go with that in a second. So, you know, using a systems biology approach, which is what most people are familiar with, and tackling these problems is the goal. So some examples of past publications. I'm sure many have seen this cell paper. John had mentioned this in the thing. This is probably one of our similar papers that involved many people. This is part of a bigger package that me and Chris and Susan Bailey had put together and Cell Press back in November 2020 that involved a lot of different papers, some from the AWGs, some outside of the AWGs. But this is people involved. So you can see many people from the AWG and people from our AWG brought other people involved to be part of this paper. So, you know, it's just the network grows as we branch out. But this paper, I think, was a seminal paper that we put out that within the group that showed that mitochondria was a key regulator in spaceflight, looking at many different GeneLab data sets. And then we ended up also getting from Chris the twin study data to add that as a confirmatory step. And then also from Scott Smith, they're not in the AWG, Brian Cruz and Sarah Zwart, but they're collaborators. So they're at JSC. They provided this physiological data from astronauts, like 59 astronauts. So he had a lot. It was a very impactful paper, and this is something that could be an example of what we've done. There's other papers, like this one is another paper from AWG's members that we put out looking at different genome data sets and looking at radiation and ground studies. These are the people involved, not as a big team, but still a pretty nice team, and we put together a very nice paper there. So now we're not worried about the past because you guys could read all those papers and see what we've done. There's other papers too that AWG members put out, but just highlight those two for now. But what we have submitted this year so far. So there's a lot of neat projects. So you're going to hear Jean talk later today. She's from University of Malta, but this is a very neat paper that she had come up with the idea saying, you know, what's the risk of gynecological cancers in space? I'm like, well, that's a very understudied field and very important study field that we should actually look into. As you probably know, in GeneLab, there's not any data sets with gynecological cancers, but you can get creative and look at different patterns. So this is a two-part project. The first part is a review with many different AWG members. The green highlighted boxes are from our AWG, and then the black one, the non-green ones are people brought in from various collaborators to help out too. But actually, we just heard this morning from a reviewer. We got probably one of the best reviews I've seen. Two reviewers said no changes, and the third reviewer said do some minor changes, and the editor thinks pretty much going to be accepted. So then we've got some good news. This paper is going to be accepted. But it's a nice review paper talking about gynecological cancers and how spaceflight could impact this. And Jean's going to talk all about it later today. So stay tuned to that. There's another nice AWG paper led by William and Rebecca. Rebecca's going to give some lightning talk, so I'm not going to go into too much detail, but you're going to see her talk in a little bit. But this is a nice other paper led by AWG members and included AWG members where William was looking at and Rebecca were looking at key spaceflight differences. This one's under review. I think it should be accepted soon too. They're addressing the reviewer's questions. But they're looking at strain differences in the kidney and then how that might be different and what people should pay attention to, and might set precedent, as Rebecca's going to talk all about it, about future experiments when you're thinking about kidney in spaceflight between mouse strains. Here's another paper, again, led by William. William was a busy man this year. And then, but the lead author was Geraldine, and she was looking at the muscle and liver, liver and muscle, or muscle liver, across differences in metabolism and related metabolic stress. So this is a very nice paper that's under review right now. Hopefully should be accepted soon too, where she did some nice, the team did some pretty nice analysis to look at different liver and muscle key differences and similarities that are related with metabolic processes. And then this is a paper that was led by Leah, but this is also Richard Britton he's not part of the AWG, but he's a collaborator of many people, including myself, Leah, and many other people, probably at our AWGs, and he brought in some proteomic data for the AWG to help. So I asked him, like, if you have this data, why don't you bring it into our AWG? So he presented, and many people, these are the people who hopped on and helped. Mark and Bill are not part of the AWG, but they're also contributors to this paper. But this is a very nice paper. It's submitted. It's under review currently. So hopefully we'll get some good positive feedback and it'll be accepted. But basically looking at rats that Rich had exposed at Brookhaven National Lab with their this kind of radiation silicon. And so ground simulated experiment, and then took out the brains and then looked at the medial frontal cortex, and then ran some proteomics, and with the help of many nice people at the AWG, came up with a pretty nice story of what's occurring there. So stay tuned for that one. So now ongoing projects, and there's a couple of other papers that I'm not going to highlight here, but as for the sake of time, this is it. You're going to hear Henry Cope talk in a little while for the lightning talk, and he's talking about the skin changes. I won't go into that, but he's another AWG member working at the AWG folks. So now ongoing projects of papers that will be, and I'm not highlighting every paper, but that's going to be probably published in the next few months, or submitted, I should say, in the next few months. One is this ongoing project. I put the key people involved running it, but there's actually 25 plus AWG members involved. So now that paper we put out in Cell, looking at specific mitochondrial changes, that was a kind of a rodent specific and human specific study. Now, I think it was Richard Barker who came first and said, hey, there's some neat mitochondrial changes if you actually look at it in plants. So then I'm talking to various folks, said, let's just make a massive effort to look at every AWG GeneLab data sets, and then also then look at the specific mitochondrial analysis. And so far, it's getting some pretty neat results, looking at how plants, Drosophila, C. elegans, microbes, and so on, have some interesting mitochondrial metabolic similarities. And Tin is really, and his graduate student Fee is really leading some of this with his tool, CPA tool, which some of you may have heard of, that he published last year with a few folks that are here. And he's using that tool as for pathway analysis, where you're making custom kind of mitochondrial pathways using Mitocarta, a broad resource. But then you can customize a pathway and you can run it through different GeneLab data sets. And this is just an example of what's coming out of this. These are mito pathways, so Mitocarta related pathways like oxphos related and other metabolic pathways. And this is just looking at specific rodent ones. He's just recently looked at plants and other things too, which we're seeing some interesting responses. For example, the thymus, which we didn't analyze the data set. The data set wasn't available for that Cell paper. Now there's some interesting response. You can see a universal downregulation of mitochondria, for example, the thymus. And then you correlate this to the plants and other data sets that we're all looking at. So this is a really exciting project. The more the merrier, so more people want to join, can't help. Big Boom, she's at a hospital in Florida, Brandon Regional Hospital, but she's also looking at the impact of spaceflight on female reproductive systems. That's an important question to ask. So she's looking at a project called estrogen, looking at basically estrogen and insulin resistant signals, and other AWG members are trying to help her out on this now, but more the merrier in this one too, because we need help analyzing some of the data sets and looking at, she's already had some interesting results she's been presenting at different meetings. And then another nice project, is this frailty project. I don't have a picture of Andrea, so I gotta get a good, nice picture to put on there. But he's actually waiting to hear back from some graduate schools. So in our AWG status of what your career level is, whether you're a professor or a director or a student, doesn't discern on if you have a good idea, we promote it and people help. So Andrea is one example of that. He came up with an idea of looking at frailty in the clinic, since he's working in the clinic. Is the idea of like how you judge your, for example, health risks happening with age. Okay, 2 minutes left, almost done. So then, he said, what about having him coming up with a frailty index, in spaceflight? So when you're in space, can you have a marker as you're monitoring to say, something's happening where you've reached a frailty point where it's, where you should start thinking of mitigating that. So for example, like causing, in this example here from this publication, comorbidities related to like increased inflammation, things like that to impact your heart. But, this is not related to spaceflight. So a bunch of AWG members, and now all of these people are involved and helping him out. And we're trying to work on a paper to get out probably in the next 2 months. And then another follow-up paper after that. And then there's another nice paper led again by a student, Amen. He's at University of Aberdeen. And she's interested in looking at ethics of genetic modification. So this is, I think, an important question that, of course, happens in the clinic. And this is some of the nice papers and articles and news articles talking about, in the clinic, how genetic engineering and the ethics behind it. But no one's thinking about that for space. And this is probably something we should start thinking about. So he's leading that, and a bunch of AWG members said, oh, yeah, we'll help out. And so we're looking at that and how to, he's already, the paper is well underway and looking at, because it will be a thing to think about, genetic engineering spaceflight, and might as well tackle the ethics now. So a nice blueprint that. So that's it. So no, this is some of the projects. There's actually probably 4 or 5 other projects people are starting to brew and work on, but don't have time to go on it. So many different projects. The more people there, the more projects we can do and work together. And yeah, and hopefully this map keeps growing to cover those other states that we don't have any members from, and other countries. So thanks, and I'll stop there. Stop sharing my screen.
Thank you so much, Achin, for that update. So the next speaker we have is Dr. Colin Cruz from Los Alamos National Labs, and he will be giving an update on the plant AWG. Take it away, Colin.
Here we are. All right, can everyone see the screen?
Sure can.
Awesome. So I guess I do want to start by thanking and applauding Kate. It's definitely, no matter how much space research I do, I will always fan over our astronauts, and getting to encounter somebody who's such a brilliant astronaut and scientist is just a privilege. And I also want to thank, of course, Lavorka for putting this whole day together. She's been Herculean in all of that. And I guess last, I do want to thank the Los Alamos National Lab for letting me maintain my leadership of the plant AWG, despite not having active NASA funding. So I really have to thank the Los Alamos National Lab and my team for making that possible. So I will say, because Sarah's talking, Rich is talking, Emma's talking, and Pankaj is talking, a lot of the highlights that I would normally be making, you'll see in other talks. So I'm not going to dwell too much on those, but definitely I'm really impressed and honored to get to work with all of them. And so I guess I'll talk more about the objectives and motivations of our plant AWG. One of our primary directives is to develop new collaborations. I'm really happy that we've been able to connect Dorothy Shippen to the Wyatt lab to sort of initiate a telomere research collaboration, because there happened to be just enough extra tissue to make that possible. So kind of look at the plant side of our telomere maintenance in microgravity, and a number of other collaborations that I will kind of talk about briefly. Of course, defining kind of the directions and potential for genetic interventions in our plants is in many ways our primary directive. We have a lot of Arabidopsis data now, and of course we need to start moving into other plants, and we need to start sort of intelligently engineering how to make those plants serve the needs that we will have for long-term spaceflight missions and inhabiting other planets. And of course, we've certainly run into obstacles that have really pushed the need for improved metadata standards and other kind of standard setting to really enable open science and make sure that we have the longevity of these data sets and they keep serving the scientific community kind of in perpetuity. I will highlight kind of some of the most active members, and I do want to highlight, of course, one of our newest members, Dana, who's going to be giving a talk at the British Society of Philosophical Science, and so she's going to be discussing a lot of very interesting epistemological standard setting to enable open science to be more effective, and that's ways that we can reduce sort of experimental biases across data sets, and really making sure that whether you're running your data on a Hi-C or a MySeq, and who did the library prep, how are we doing standard setting, and what protocols can we initiate to make sure that those data at the replicate level are comparable? Because as I think many people probably have noticed when analyzing GeneLab or other archive data, is that we end up hitting places where we really can't compare the direct replicates of experiments, despite being able to kind of compare common changes, as I think Dr. Wyatt exemplified very, very well. Of course, I encourage you to stick around and make sure that you get to see Richard's talk. One of the first and primary efforts of our AWG has been to look at the existing data sets, so 15 different spaceflight transcriptomes, and really identifying what are the true norms of the microgravity response, and what are the unifying responses that we can tweak or modify to make sure that we are understanding plant responses and enabling the transformation of those responses to suit our astronauts and long-term spaceflight needs. I do want to highlight Emma's work. She has done a major undertaking, read over 200 primary articles, identifying 78 specific loci that have gravity defects in the Arabidopsis genome, and some outside of Arabidopsis as well. And really then taking that and going beyond it to look at gene interaction analyses to identify some truly promising targets for genetic interventions. And I think the work that she's done with, guided by kind of Dr. Wyatt and Dr. Christina Johnson at the KSC, will really form a great roadmap for genetic interventions in the future to try and, again, improve plant performance in the microgravity environment. I did want to also highlight one of the more interesting trends evolving as we've been getting fractional gravity data coming out of spaceflight missions. And that's, you'll see kind of our microgravity, our moon, our Mars, and our reduced G, and then all the way up to our kind of our full 1G. And so these are all, each one of these squares is a cluster looking at a set of genes that are moving in similar patterns across different treatments. And then the number of genes on that top right there, and then the trend of differential expressions in each individual heat map. And what you'll see is that there's a very different trend that kind of starts evolving at the microgravity level, but we also see a similar set of genes that are moving in similar ways, whether once we go above a moon, Mars, and reduced gravity level. However, when we really look at our very low G's, so between our moon and our microgravity, we do see a very distinct response. And so we're going to have to be very careful, and this is definitely a gravity, a fractional G level that needs to be more investigated to try and tease apart what we're going to deal with in the sublunar gravity levels. And so this work was done by Raul Herranz. It's really fantastic work. It came out in Frontiers already, but I will give you a little teaser and say that there's going to be a great deal more kind of mutant analysis and follow-up work coming out of this to really show what is happening here. So kind of, I hope you're all on the edge of your seat for that, because Raul always does some really amazing work. And one kind of big takeaway of this is, again, we are seeing over and over again the interaction of light and gravity across these data sets. And at that very low G, we are also seeing a major distinction in how the interaction of phototropism, so response to light, growth response to light, versus gravitropism, so that response to gravity, how those different stimuli are interacting. And so definitely there's a interesting sweet spot between microgravity and lunar gravities that has a unique and distinct response. And so that's going to be an important area to look at in the future. So a lot of work has been done out of the AWG to do that. That's of course been led by Raul. So if you have more questions, certainly pick his brain. He is a great force in this field. Oops, sorry. And then I do want to also highlight just some of the exciting things. And I know that this is not exclusive to our AWG, but Mark Settles, one of our members, has recently gotten a really exciting new development where one of his hardware priorities was set for the Artemis science. And so he'll be doing yeast grow-ups on the moon that kind of get to follow in line with some of his algal work, looking at DNA mutation rates outside of orbit and microgravity aboard the ISS. So I think this is going to be one of those fundamental pieces that we're going to have to look at to understand the full extent of how radiation is going to affect our plant systems, but of course, our human and bioproduction systems beyond that. I may have just breezed through that, but that was kind of on purpose because I do want to leave the primary stories to Pankaj and to Dr. Wyatt, and of course to Emma as well, and Richard, who's going to highlight kind of that primary thrust of our work. And with that, if there aren't, I guess this is the end of the session. I don't know if we take questions now or if I should hand off anyways, but thank you so much.
All right, thank you, Colin. We have our final speaker of the category, and that is Ryan Scott. He's from NASA Ames Research Center, and he will provide a status update on the ALSDA AWG. Take it away, Ryan.
Great, thank you, Lavorka. Can you see my screen and can you hear me?
Sure can.
All right, thank you so much. So I do want to just, the ALSDA stands for the Ames Life Sciences Data Archive. And the same way that the AWGs were started originally to help standardize metadata and processing and the database standards to enable this community to mine the data, that's where ALSDA is at now. And so I do want to give a bit of an update and first sort of talk about why is data sharing so important for the community on a bigger scale. That is knowledge discovery, of course, using bioinformatics and systems biology. Meta-analysis, of course, is another approach and for reason to share and curate good, strong scientific data. Knowledge graphs and applications also to machine learning are also all burgeoning and really important new ways that data can be used towards knowledge discovery. But data sharing is also going to be important for future ecosystem development and the various different platforms and hardware tools to either support the ecosystem or to have experiments on the lunar, Mars transit, Mars surface missions. There's some really good new publications sort of on this topic that I have cited on the bottom right. Lastly, one of the other important things is thinking about the big term. If there's a plant or a yeast or rodent experiment on the lunar surface, it's important to involve the country and the world in that effort. And data sharing is a really important way to involve the world, also build confidence in science, have transparency in science, and data sharing is key to enable a lot of that open science approach. And the last thing I do want to talk about in terms of data sharing is also taking translational data from model organisms to support human health risks. And so there's a outfit called the Human Systems Risk Board, which is centered at Johnson Space Center. And they've created what they call directed acyclical graphs. And these are risk modeling tools to be able to map out how microgravity leads to bone resorption, which leads to urinary calcium excretion, which leads to renal stone development, which leads to the increased chance for urinary tract infections, for example. All right, but the reason and what the Human Systems Risk Board has done has actually then created these DAGs, because that doesn't live in a silo, that microgravity to UTI. That is, when you have SAS, space adaptation syndrome, oftentimes anticholinergics are prescribed, which leads to urinary retention, which is then you have a catheter usually introduced to relieve that, which changes the probability of UTI, right? So you can see, and then this all eventually leads to sepsis, or maybe negative crew health outcomes or negative mission outcomes. And so just another example of another reason that we need to collect data in a way, and that all biomedical data should be seen as precious, absolutely precious national and international resources. So ALSDA is joined with GeneLab last year into really one overarching project. And when you think about biological data, think about the multi-hierarchical level, right? Of course, the genotype and what GeneLab has pioneered in the last several years is absolutely stunning and impressive. And what ALSDA has been working on since it started this effort to modernize last year was begin curating and standardizing all the phenotypic level, the physiological, the behavioral, any pathological outcomes, and all the different formats, not just tabular data, but also bioimage data, video data, and most notably mission telemetry data, right? This oftentimes, a lot of times when missions are sent to space, how they're going to curate and standardize data for long-term use for all time is oftentimes a overlooked fact. And so mission telemetry data is part of this. And once all of these levels of data are able to be mined together, the ability to have multi-hierarchical, multimodal data for various computational informatic approaches is really exciting over the next decade. And so in the last year with ALSDA integrating with GeneLab, this is going to enable a single submission portal. So regardless of what type of data, biological data type you have, you can go into the new portal, which is ALSDA and GeneLab, and you can submit your data into collaborative workspace. There's still going to be the data portal, which the repository, which most people know. There's the environmental analysis for various environmental telemetry. There's visualization tools for statistical visualizations. There's, of course, the Omics toolshed, which most people are probably familiar with here. And the new thing that is being worked on right now is a new AI modeling zoo portal of sorts to mine various different types of data sets together, be that molecular omics, phenotypic, or telemetry all together. Right, so it's a pretty exciting things coming up. And so what this means for egress and ingress of data, in terms of the egress, it's going to look pretty similar in terms of like you're going to go to a data repository, but now you're not just going to have transcriptomic data available, you're going to have micro-CT data available, ultrasonography data available, Western blot, calcium uptake spectrophlorometry. There's many different types of data that are being worked on. And then when it comes to the ingress of data into the new data system, when a person goes to submit their data set, when you click on, right, because standardizing the investigation level metadata, the sample level metadata, and the assay metadata are all key. And so when someone goes to submit different assays, what the ALSDA has been really working on, collaborating hard and getting feedback in the last year, is pretty much adding these phenotypic ALSDA assays for PIs to submit data to. And so we've had 8 meetings starting last July. We have a roughly 100 members. It sort of ebbs and flows how much time people are graciously willing to provide feedback to. Here's a bit of a snapshot of who's in the AWG across both space biology and human research programs. This is a snapshot of what we call the assay metadata configs. That is, how was that ultrasonography conducted? What were the specifics of the probe, or the micro-CT scan that you did on the eye? Like, what were the parameters connected to the assay itself? And those types of specifics that are important for longevity and reuse of the data. And this has led to about 20 total metadata configurations, which is amazing, that we've received feedback on from the AWG community. And there are a number of AWG members have submitted data, which is exciting to have people excited to share data. People are beginning to see that there's an original research goal of their data, but there's a secondary reuse possibly as well as mission telemetry feedback. And most notably, Jaden Hastings will talk about AI readiness in her lightning talk later. So I'm not going to walk through these, but the other thing that the AWG has contributed to is the production of the database workflows. And so we've come up with the work, because we know there's going to be new assays in the future, and there's assays we haven't finished yet. So how to create a workflow that's trusted for the assay metadata configurations, when we receive a data set, how to curate that, and also how to curate it in a manner so that there's a data template of the results, so that dependent variables across every ultrasonography data set are identically the same and structured, so that it can be mined by machine readable approaches. So just wanted to throw that out there. It's in the background, but the workflow, just producing these workflows has been really amazing. Another thing the ALSD AWG has been involved in is our collaborator Svetlana Komarova at McGill has a history of running these meta-analyses looking at bone. And just in the last several months, a new cardiovascular meta-analysis project has started with the AWG. Part of the group is doing the literature review. Another part is really going to be focusing on analysis, but it's going to be looking at cardiovascular parameters across legacy data sets. And this is a bit of a snapshot of who's involved, with also Sunny Narayan and Matthias Nesset. And interestingly, 100 young people signed up to be involved in literature mining and review of literature. And eventually what we will be leading to is actually submitting the mined literature, right? I know it's oftentimes summarized or means from these literature data sets into ALSDA, which is what we've already done with Matthias. Is there's several of these data sets that you can see here that were mined from the literature. Then the data was collected and now has been submitted into, it's not the raw data, but it is the summarized process data, which you can still get results at. And so this whole entire effort is going to be awesome to demonstrate the value in the legacy data. There's this community approach for curation is going to be amazing, and the experience for these hundreds of students across the country that signed up to be involved. And because ultimately, the more data that we have, the more reuse is possible. And speaking of reuse, it's sort of stunning how every data set that gets submitted to ALSDA in the past few years, I mean, it's stunning how many reuse examples we have. There's a microscopy data set and a micro-CT data set that are both being worked on through the Science Mission Directorate in their AI initiative to do benchmarking so that they will change, so that they can train future machine learning models. There's a federated causal inference so that, and this will help enable in situ analysis on station or on the moon or on Mars transit for machine learning applications, and protect privacy because you transfer the model, not the data. There's also efforts to look across omics to phenotypic data using machine learning. There's the meta-analysis work. There's a new explainable AI collaboration to look at the differences of fast and slow twitch muscles impact from omic to phenotypic. And excitingly, we have our first manuscript that we're submitting today, which started in the AWG last year, which is looking at bones specifically connected to the Human Systems Risk Board. So in terms of the big picture, you know, to get to the high level of machine learning type approaches, we have to have strong basics so that all data is fair and open. And in the next coming year or so, we're probably going to be focusing a lot on bioimaging data, the mission telemetry data, curating some of the legacy data sets, and ensuring that all of our data is AI ready. And so I obviously need to give huge kudos. And this is just a portion of the AWG members that have either contributed data, edited configs, provided feedback, and we have a monthly meeting, and it's been a real humbling experience to be part of this growing community. And I do want to leave people to sort of, when we conduct science and when we think about the data that's connected to all experiments, think of yourself as a citizen in time. All of these data are precious resources. Every experiment, every payload, and we all have a duty to have good data stewardship, because this literally is going to ensure repeat knowledge discovery, support ecosystem platform development. It'll enable the world to be involved and have transparency, confidence in science, and ultimately will support operational and mission health outcomes. So yeah, just want to thank everyone, and yeah, appreciate it, Lavorka, and everyone here. Cheers.
Thank you so much, Ryan. That was a great update and background on ALSDA AWG. So we have concluded this particular category. I do want to, so we didn't really have any Q&A planned, but if you do have any questions for the speakers in this category, please put them in the chat, and leads, if you wouldn't mind just answering the questions when you get a chance. And if you are interested in joining the AWGs, please feel free to fill in this form so you can be involved. So with that, we're going to take a 20-minute break and come back at 50 minutes after the hour. Thank you all. All right, welcome back, everybody. We will continue the AWG Symposium with our next category called AWG Early Career Lightning Science Talks. This section contains a mixture of graduate students and postdocs, and will highlight some of the exciting work with which the early career scientists from each of the AWGs are involved. There will be 4 speakers in this section, and just a reminder that because these are lightning talks, we will keep the Q&A at the end of the category when all of the speakers have finished. And speakers, I will be giving you 1 ding of a bell for your 1-minute warning. So with that, I would like to introduce our first speaker in this category, and that is Henry Cope from the University of Nottingham. And he will give a lightning talk about the impact of spaceflight on the skin transcriptome. Henry, if you are here, go ahead and take it away.
Okay, thank you. Okay, yeah, so as mentioned, I'm going to talk about the effect of spaceflight on skin, particularly in mice. So this is mainly supervised by Afshin Beheshti. So firstly, why look at skin in space? So for one, skin manifestations such as rashes are one of the most commonly reported in-flight clinical symptoms. A famous example is when Scott Kelly came back from his year in space. He had quite a severe rash. Skin also can tell us about the kind of whole body health, for example, the function of the immune system. And importantly for spaceflight, skin is the first line of defense against radiation and also pathogens. So of course your cosmic radiation of being in space, and then also the kind of closed environment of the ISS, potentially increasing risk of infection. And skin also presents an interesting opportunity because it's easily accessible. I mean, it's the first thing you come across. So it could be assessed with non-invasive methods. And then there's also quite a good kind of target for treatments, like topical treatments. And so I think overall skin is currently understudied in space. Okay, so the core data sets used in this project are 5 different GeneLab RNA-seq data sets of skin from mice that have been flown to space. And so these represent a number of different variables, including 3 different missions, and then different things like strains, like mice strains, kind of diet, time spent in space. So it's really important to kind of try and understand the differences between the data rather than just kind of blindly combining it all. So the kind of key questions involved with this project are what kind of responses are common across the data set? So what's the common response of skin? And then also which responses kind of seem to be different between the different data sets, so are affected by the differences in diet and stuff, for example. And so firstly, because we're doing a comparison between flight samples and ground control samples, it's important to question whether there's actually a difference between the two, whether spaceflight is causing a difference. And from the PCA plot shown here, the kind of blue samples for each data subset. So 5 data sets, but I actually kind of split them into 10 data subsets based on the kind of different conditions. So the blue samples are the space samples and the red samples are the ground control samples for each data set. These are significant genes. And there's a nice divide between the two. So that shows a difference between spaceflight and ground. Kind of using an approach like looking at pathways that are significant across most of the data sets, and then further filtering the genes involved in these pathways to find the genes that are significant in at least 2 out of the 3 spaceflight missions. We find kind of genes associated with DNA damage response, such as BRCA1 gene, and also a cell cycle. And we're trying new approaches to this kind of network analysis and stuff now as well. Another approach is that we're looking at curated kind of lists of pathways, for example, and then seeing what the response is in the skin. So here we've worked with a dermatologist to create a list of pathways associated with skin health, which are kind of shown as the columns on the left. But I'd like to kind of direct your attention over to the right side where we've got a heat map of the genes that are involved in those pathways, those significant skin health pathways. And particularly in the top right, you can see a big blob of blue, and that's a down regulation mainly of collagen genes. And it's just the top 4 rows, which are, so 4 of those data subsets. And then the ones below it are all kind of red, but not as significant. And those blue ones are all involved with the JAXA MHU-2 mission. So therefore, we can say that the difference between those that data set and the other data sets is driving a difference in skin, and that could be due to hypergravity event because they didn't have recovery time and they were returned to Earth, or it could be because they're male mice and all the other ones are female mice. There's also some housing differences. So next steps, I mentioned we're working on a more kind of sophisticated approach for identifying those common genes in spaceflight, working with that Q-lattice algorithm from company Abzu. We're also preparing a manuscript and working with the GeneLab for High School program. So they're helping, some of the students are helping with the literature review, for example. And then also a huge part of this project that I can't talk too much about in this presentation is the human data. So we've got lots of kind of data from the NASA twin study, JAXA hair study, potentially like the commercial Inspiration 4 study. And we're going to be comparing that. So basically, given what we found in the rodent data, the kind of hypothesis generated there, do they also hold true for the astronaut data? And then from there, are there any potential pharmaceutical targets, such as creams, for example, or any kind of follow-up studies we might want to do in space? That's all. Thank you very much.
Wonderful. Thank you, Henry. Our next speaker is Dr. Jaden Hastings from Weill Cornell Medicine, and she will talk about AI readiness toward applications in space biology. Just a quick reminder to the audience, if you put in questions into Conference.io, if you can please put the name of the speaker for which that question is intended, that would help me. Thank you so much, and take it away, Jaden.
Thank you very much, Lorca. Thank you all very much for your attention. So I'd like to take this opportunity to share a little bit of a summary of some of what's been happening this past year concerning questions around AI readiness of our data that's within NASA GeneLabs. So in the spring of 2021, the attendees of 2 workshops were tasked with the development of a series of recommendations for the adoption of machine learning and AI, or augmented intelligence, for space biology and astronaut health in the support of NASA's decadal survey. The effort to produce the concrete recommendations for further action was led by Lauren Sanders, Ryan Scott, and Sylvain Costes, who are amongst us. In addition to this white paper, NASA submitted to NASA for consideration was a workshop summary report sent to NASA leadership, as well as 2 review articles currently under review in Nature Machine Intelligence, for which I'm one of the co-authors, and currently available as preprints, which I can make available later. So from these discussions, the notion of a data lifecycle and the need for a data management environment emerged. And within this DME could be embedded data retrieval tools, ontological mapping capabilities, meta-analysis tools, and data visualization and modeling tools. And the investment towards the development of this infrastructure would allow this research community to aggregate and integrate multiple data types, like biological phenotypic data, various molecular omics, physiological, biochemical, or imaging, qualitative behavioral data such as wearable tech, textual analysis, or video, even a vivarium, and contextual mission data, including radiation, exposure, telemetry, acoustics within the environment, vibrational information, gas mixture, and humidity. So the ability to fully integrate such multi-layered data with increasingly automated biomedical monitoring systems of crew health and the development of precision space health system is kind of where we're going, we're envisioning as the next iteration, or where we should be moving. And such a system would monitor, aggregate, and assess biomedical status with increasing autonomy. The system could also analyze and predict personalized adverse health outcomes in the crew, adapt and respond to new data that is capable of providing actionable insight to deploy suitable countermeasures to maintain optimal health in the space environment, especially as crew move further out and will require additional autonomy or increasing autonomy. So the ability to automate experimentation in space could also enable self-driving labs or automated experimental platforms that can expand our understanding of the effects of spaceflight on living systems while decreasing the demand for crew time on orbit, which is very expensive and precious, as we know. Automated and AI-assisted data harmonization and dataset curation is critical to advance space biology research within this architecture and can support the entire experimental process, from investigation management to experimental execution, data publication, and even preliminary analyses. The investigators on Earth or elsewhere can then utilize an embedded digital experiment notebook to preserve experimental parameters and analyses with link out capability to approved external data repositories. The key here is increasing automation and hands off humans out of the loop experimentation. So key to the deployment of ML and AI within the domain of space biology are a set of immediate priorities to address, which include community capacity and consensus building, both within the research community and with new and emerging stakeholders within the commercial sector, broadening access to computing resources suitable for machine learning and AI in a manner that is adherent to FAIR, or the principles of FAIR, findable, accessible, interoperable, and reusable, as well as open source principles. building structured data repositories that improve reproducibility and therefore build trust and AI readiness, which leads me to our final call to action. And this is in collaboration with our amazing colleagues over at NASA GeneLab, and a particular shout out to Lauren and Ryan. Up for consideration amongst this group is the formation of an analysis working group comprised of subject matter experts that are committed to the development, so this infrastructure and capacity building of data sharing standards to ensure that this data that is currently held within the GeneLab repository, as well as its other associated repositories, is AI ready. So we'd like to extend this invitation to all of you to participate, and I've included a QR code for you to scan and then link through to the Google form if you're interested in becoming a member of this AWG, or would like to simply be kept in the loop of any developments in the future. Thank you.
Wonderful. Thank you so much, Jaden. The third speaker in this Lightning Talk category is Rebecca Finch from Staffordshire University, and she will talk about exploring kidney dysfunction in space, comparing responses of mouse strains using omics. Rebecca, if you're on, go ahead and take it away.
Thank you very much, Livorka. And now, as Livorka said, today I'm going to be talking about my master's research, which explored kidney dysfunction in space by comparing responses of mouse strains using omics. Now, microgravity and increased radiation exposure in space leads to oxidative stress and mitochondrial dysregulation. The mitochondria can deal with reactive oxygen species, but when overloaded can become dysregulated. Now, the kidneys are very rich in mitochondria, so they are severely affected by oxidative stress and also very important for preventing it. Renal fibrosis is a disorder associated with oxidative stress in which overactive wound healing is triggered by an initial injury, for example, a kidney stone. And a major component of kidney stones is calcium. Microgravity and increased radiation in space can lead to bone loss, which leads to...
Can I stop you quickly? You're not sharing, we're not seeing your presentation.
Oh, I'm terribly sorry. Let me share. Let me just share my screen. Sorry about that. Okay, are you now able to see my screen?
Yes, perfect. Thank you. Oh, you went back to your slide deck here. Yes, there you go.
Oh, yes, thank you. Okay, so sorry about that. A major component of kidney stones is calcium, and microgravity and increased radiation in space can lead to bone loss, which leads to increased calcium levels in the bloodstream, which leads to increased incidence of kidney stones in astronauts. Now, mitigations are in place, but currently are insufficient to limit the risk of kidney issues on long-term missions. This can impact the health and well-being of astronauts and limit their productivity, and in a severe case may cause the need to abort the mission and return to Earth for treatment. So the aim of our research was to determine if genetic differences have an impact on the response to spaceflight. So we used transcriptomic data sets from two different rodent research missions, RR1 using C57BL6J mice and RR3 using BALB/C mice. The two mouse strains are genetically different, and BALB/C mice have been shown to react less severely to kidney injuries. So in each mission, a group of mice were flown on the International Space Station. Another group were housed in the same conditions on Earth to serve as ground controls. RR3 also used a basal control group, which were sacrificed on the day of launch. So at the end of the mission, the flight and the ground control mice were sacrificed and omics data obtained and uploaded to NASA's GeneLab platform. And then we used a number of methods to analyze the differences in gene expression in this data. So the heat map for C57BL6J on the left shows a strong separation between the genes differentially expressed in the flight and the ground control groups. The heat map of bulb C in the middle shows a weaker and a more subtle signal with less separation between the groups. Now, a comparison of the expression patterns of gene networks between the two strains on the right identified clusters of gene sets exhibiting overrepresentation with opposite patterns. Now, cholesterol and acyl-CoA biosynthesis were positively overrepresented in C57BL6J, while negatively overrepresented in bulb C. And the opposite pattern was seen in clusters related to translation, protein folding, and circadian rhythm. So, but not shown here, but also lipid metabolism, cell cycle, and immune response processes were positively overrepresented in both of the strains. So the bulb C mouse strain has a distinct set of genes with non-synonymous mutations compared to C57. We analysed these genetic differences and found the hyaluronan metabolic pathway had the highest enrichment ratio, and genes in this pathway were differently expressed by bulb C mice in spaceflight. So to investigate the potential effects of the mutations, the differentially expressed genes in C57 were filtered by only those which were mutated between the strains, which is shown on the left here. And these included genes related to cholesterol and fatty acid metabolism, cell junction, and PPAR signaling. The same was done for the bulb C differentially expressed genes on the right, showing genes involved in gap junction and lipid metabolic processes. So in summary, spaceflight has a significant effect on gene expression in the kidney. Bulb C mice were shown to have a milder response than C57BL6J. Now, we found signs of reduced hyaluronan synthesis in bulb C, which could possibly be protective against extracellular matrix remodeling. More research is required to confirm this, but identifying genetic targets related to the development of fibrotic kidney disease and using these to develop new drugs could potentially mitigate kidney health risks to astronauts and also help people on Earth with kidney disease. Thank you very much for your attention.
Wonderful. Thank you so much, Rebecca. And our fourth speaker is Emma Kennedy from Ohio University, and she will talk about mining the gravity mutants of Arabidopsis.
Are my slides visible in my audio all right?
Yes.
Great. So I am working on a review paper that's looking at mining the gravity mutants of Arabidopsis and how those mutants and ground research can be used with the GeneLab data sets to really better understand the core mechanisms behind gravity signaling and perception. So first off, if my slides will progress, what are gravity mutants? These are mutants with the breakdown in specific sense, signal, and response to gravity. So as we can see, if you grow a plant and you put it on its side, wild type plants will bend so that the stems are going up and the roots are growing down. Gravity mutants will have one of these major 5 phenotypes where either they're reduced and don't reach that same degree of curvature, they're delayed where they take a longer time to reach that degree of curvature. Overachievers will do something really strange where they bend past the new gravity vector. Reverse mutants will have a stem or a root that's going opposite to its normal direction, and agravitropic mutants will grow completely outside of their gravity vector. They don't really respond that much to orientation because they weren't growing with gravity in the first place. So looking at a review of over 200 primary publications, we've developed a list of 78 loci that are gravity specific. From the phenotypes, we can see that the majority of those mutants have a reduced phenotype. So even when you knock out, say, an entire gene family, there's still some level of gravity signaling getting through and being transduced, and the plant is able to respond. We also see that through perception, signaling, and response, signaling is by far the most diverse category. There's gravity mutants that are involved in protein modification, environmental responses like heat shock proteins and reactive oxygen species, and quite a few in cellular organization, membrane-bound proteins, vesicle and vacuole modifications, and of course, signaling molecules. We can see that there is a lot of information on these mutants, but they're not necessarily all that well connected right now. there's a lot of papers have referred to this as the gray cloud of gravity signaling. There's a lot of information. There aren't clear signaling pathways. So what can we do to take that in the future based off of this mutant list and develop a more cohesive model for gravity signaling? The main thing that we advocate for in this review would be the production of new mutants. So taking this list of gravity loci, we can use tools like KNet Miner, which take a list of known genes and return the potential known interactions. So for this gravity mutant right here, like SGR7, we see that it has 2 known substrates and 1 known activator. So if we could produce mutants for those known loci that are interacting with gravity mutants, we might be able to pull out more cohesive signaling pathways as opposed to individual proteins and transcription factors that are involved in gravity. Another really amazing thing is that most of these mutants, if they have any molecular or transcriptomics data at all, that is GeneLab based data. So even taking the ground control studies for these papers that look at mutants and wild type on flight, if we look at the ground controls and just compare the mutant transcriptome to the wild type transcriptome, we can pull out the phenotype and mutant specific changes within those ground controls and see what pathways they're truly involved in. Together, by combining these gravity specific mutants and using those with the GeneLab transcriptomics and proteomics data, we can combine the last 50 years of ground-based reorientation studies with all of the cutting edge data on GeneLab and really get a better idea of the larger signaling pathways involved in gravity perception. With that, I would like to thank all of the plant AWG members, especially those that suggested mutants and gave me information about the mutants that they work on in their labs, especially Dr. Christina Johnson and Dr. Sarah Wyatt for their guidance throughout this project. Thank you.
Wonderful. Thank you so much, Emma. So we're going to have a couple of minutes here for questions from the lightning talk speakers. And our first question here is for Henry. Henry, if you wouldn't mind turning on your video. Are there any effects to understand 3D printed skin in the future as well? ESA is about to publish grant opportunities, and it looks like the skin is one of the most desired first approaches.
Yeah, okay. Thank you very much for the question. So I did see, I think, because we did quite an extensive literature review for this project. And so one of the things I came across was a perspective article. I think the authors are from DLR maybe, or another European institution. And they were talking exactly about that as a potential thing, 3D printing the skin for spaceflight. And in general, kind of sending up those fibroblasts and things like that is something that there's been quite a few European studies on. So yeah, I think that is quite a good candidate for bioprinting, and I could see that kind of being quite a popular area in the future.
Wonderful, thank you. Our next question is for Jaden. Will AI change automation on the ISS? We are thinking to give an AI approach a shot to decide if the experiment should be treated during spaceflight. Is anyone implementing this already?
So, to my knowledge, there is no human out of the loop, but rather human in the loop application. And if you'd like, you could send me an email and we can follow up on specifics of experiments that at least use machine learning AI as a different question. Right now, compute power is one of the limiting bottlenecks for deployment of machine learning or AI. So in the future, it needs to be a bit more of a concerted effort, but we are at least inching towards deployment on ISS and upcoming commercial stations.
Sounds good, thank you. And our question here, one more for Rebecca, is were strain-specific references used for the analysis pipelines?
Okay, so yes, what we did, we took the mutations that were known between the two strains, and we looked at only the non-synonymous ones, so the ones that would make a difference in the phenotype, and then we compared those to the differentially expressed genes in space. So that's how we incorporated that.
Wonderful. Thank you, speakers in the lightning talk category. Those were amazing talks. That's a lot of information to pack into 5 minutes, so those are the hardest talks. All right, so moving on to our last category, the AWG science talks. In this section, we will have a mixture of research presentations representing each of the 5 AWGs. There will be 5 speakers, followed by a 3-minute Q&A at the end of each speaker presentation. And the first speaker in this category is Dr. Richard Barker from the University of Wisconsin at Madison, and he will be presenting about the matrix meta-analysis of the first 15 astrobotany transcriptomes. Take it away, Richard.
Hello. Can everybody see my slides and hear me okay?
We can see you. Not sure what your slides are. We don't quite see them.
Okay, 1 second, please. I'm terribly sorry. Share my desktop. Share that. Okay, cool. How about now?
Perfect.
Okay, cool. Well, I'd like to start off by thanking the committee and for giving me the opportunity to share some of the research that I've been performing on behalf of the NASA Analysis Working Group. It's a project that we call the GeneLab Plant AWG Metadata Matrix. And so this is a meta-analysis of the first 15 spaceflight transcriptomes. And hopefully what I'm going to do is give you an insight into what the normal wild-type response of plants is to spaceflight based on the Arabidopsis response. And these pictures at the bottom were made using DALI Mini to try and paint a picture of what a plant matrix might look like in people's imaginations. So Arabidopsis thaliana is a really fascinating model organism. It's exquisitely sensitive to its environment. It uses gravity to guide its roots down. And then when it encounters barriers, it can use the concept of thigmotropism to avoid them. Ultimately, the root is trying to find water to help fuel the plant and help it grow. And so that's a concept known as hydrotropism. So these early data sets, we're going to be looking at the different organs and primarily the roots and shoots. In future, hopefully we'll have access to data related to flowering and inflorescence growth. And each of these different organs, it's important to note, have evolved to sense different environmental stimuli in different ways. And that's going to become quite important when interpreting these data. So thanks to the NASA GeneLab program, there is a large menu of data that is now available for us to mine and look for coherent patterns to try and help us move the field forwards. GeneLab have quite nicely organized it, giving each experiment an accession number, which makes it easier for the community to discuss each of these studies. In addition to that, they have linked this GeneLab ID to the experimental platform. That's the hardware that the organism was grown in. They provided a summary of the factor that the research group were investigating in that study, and also listed the material type that was harvested that's available for analysis. There are many different pieces of hardware that were available, and essentially we down selected that list on studies that had 3 replicates or more. And that narrowed it down to these pieces of hardware that we use to fly either Arabidopsis seedlings or cell cultures. The classic workhorse for NASA is the brick. The brick hardware is awesome. It's nice and simple. 60 millimeters Petri dish fits into a canister. BRIC is obviously a NASA acronym, biological research in canister. And here you can see Reed Wiseman injecting RNA later fixative into BRIC-19 before those samples were turned to Earth for analysis. But for me personally, my favorite way of growing Arabidopsis is in a square Petri dish. This is the one that we use on Earth in the lab all the time. And there are some studies that have used this particular science carrier to investigate how Arabidopsis responds to spaceflight. One group used, as part of Project Kara, they were able to stick these Petri dishes to the side of the space station wall in both light and dark. Equally, the apex stages came up earlier in Dr. Wyatt's talk, and this system also used square Petri dishes and provided lights and photography on orbit. The veggie grow system is my personal favorite way of growing plants in orbit because it allows us to create a bridge between the fundamental genetics we do in Arabidopsis and the future agroecosystems that are being tested in the veggie by NASA. I'd also like to point out the European Modular Cultivation System and the Shenzhou deep flyer that flew the European Symbox. These 2 advanced pieces of hardware have centrifuges on orbit, so not only provide data about organisms in microgravity, but also provide insights into the plant response to fractional gravity levels. So essentially, we took these data, the 15 studies that passed the replication criteria and were processed by the GeneLab pipeline, the approved pipeline that was published by Overby et al. And then we were able to examine over 105 different comparisons. These data were presented in TOAST, it's a test of Arabidopsis space transcriptome, an interactive relational database that we published by Quartal 2020. But more importantly, we then went on to perform a clustering analysis using KEGG, Ensemble, Reactome to provide insights into some of the genetic clusters that are responding in similar and different ways in these studies. I'd just like to shout out about the Metascape software. It's great, provides lots of new insights and a great tool for these types of meta-analysis. This graph was presented earlier, but so I can be quite quick with it. But essentially, what I'm trying to summarize here is we had those 15 studies and the different major metadata features, and we could assign links between the different assay types, via RNA-seq and Microarray, to the different organs that were harvested in the different flight vehicles. There was a clear bifurcation of the data. Microarray and RNA-seq are distinctly different assay types. You can visualize this distinction in a range of different ways. Here, using principal component analysis, we can see the Microarray data clustering really far away from the RNA-seq. When we removed that principal component and we looked at principal component 2 and 3, essentially biological patterns started to pop out. And this for me was really fascinating. We saw the light samples on one half of the plot and the dark on the other. The major feature that caused the clustering is actually the combination of the ecotype and the tissue. And it's kind of hard to separate those things because certain hardwares only provide certain types of tissue for analysis. So with this in mind, essentially what we then did was separated the RNA-seq and Microarray data and used Wagner, weighted genome correlation analysis, to perform an unguided clustering, which separated out the gene expression patterns into groups based on both the expression of the gene, be it up or down, and the ontology group that it belongs to. Essentially what this revealed was that RNA-seq data was a bit richer. We had 4 distinct clusters in that analysis, whereas in the Microarray analysis, we only had 3 distinct clusters. The circus plot in the center illustrates the connections between these 2 distinct assay types. So essentially, this dark line here shows that within this brown cluster from RNA-seq, we saw a group of genes that were also differentially expressed in this red cluster from Microarray studies. The light blue connections show genome ontology groups that link various different loci. And essentially what this graph reveals is that there isn't a huge conserved overlap in genes between the different studies, in genes responding to spaceflight in the different studies, but there is a consistent sharing of different ontology terms, different functional descriptions that are altered in spaceflight. So in order to explore these functional factors, these functional roles, we wanted to examine the different pathways that they're involved in. I'm just going to focus in here on the KEGG pathways. That's the Kyoto Encyclopedia for Genes and Genomes. And essentially, I've selected a set which were identified in both the RNA-seq datasets and also in some of the Microarray datasets. One of the things that we can see is that the primary carbon metabolism is significantly altered. This is partly because of changes in photosynthesis and partly due to changes in oxidative phosphorylation. For those of you that aren't familiar with those pathways, essentially photosynthesis is where the Calvin cycle integrates carbon dioxide and water to make sugars. These sugars are then passed to the mitochondria, where glycolysis releases them, and the citric acid cycle helps produce ATP, which fuels a range of different processes in the cell. So this overall system is altered in spaceflight. So in other words, photosynthesis is adjusting, respiration is adjusting, and these 2 changes to how the primary carbon moves through the cellular metabolism can be observed in pretty much, well, in most of the pathways, that there are broad changes to the fundamental carbon metabolism in plants in spaceflight. Looking at these, what we started to realize was there was 3 distinct groups. We could see samples from flight hardware, such as the Kara and the Veggie, appeared to be clustering together. And that was leaded us to realize that these particular hardware types enable the samples to be open to the ISS atmosphere. So they essentially get the elevated CO2 that is present on the space station. Whereas the brick and the future brick LED hardware, these tools are triple sealed. And so that basically leads to a progressively increasing hypoxic environment internally. and that can be detected in the transcriptomes and the changes in the carbon metabolism. We then have the EMCS and can make predictions based on the APH, as the ethylene removal systems that these hardware have are also likely to alter this fundamental carbon metabolism, which will impact on hormonal movement. So revisiting those data sets and trying to illustrate the connections, what we realized was it was actually really important to demonstrate the hardware as like the core part of the different studies, and then look at the patterns that we could find within individual pieces of hardware. Okay, so far I've been trying to tell you how different all the different pieces of hardware are and how that influences like all the different pathways that are responding in them, and essentially that showing that each of the different studies is quite distinctly different, be it because of the hardware or the tissue type that was available. But what I also said at the beginning was I wanted to try and show you what the wild type response of Arabidopsis is to spaceflight. This is actually a harder question than initially sounds, because depending on where you come from, the concept of what the wild type of Arabidopsis is can be different. Columbia Arabidopsis comes from Missouri, comes from America, and yet there's varieties of Arabidopsis from all over the world. So we did a survey within the analysis working group, and ultimately learned that Columbia wild type is the standard. And here you can see time-lapse photography of that growing on Earth and on the International Space Station. These samples were returned to Earth and RNA-seq was performed, so we were able to analyze their transcriptome. We then used a multi-layer stress pattern recognition system that used 85 distinct microarray studies from GEO to create a hidden processing layer known as physiospace. This then can be used as an autodecoder to identify 20 distinct stress groups, such as drought, heat, radiation, hypoxia. And so we then applied this stress autodecoder to this RNA-seq data generated as part of APEX-05. What we can see when we look at the roots and shoots separately is distinctly different, but kind of similar results. And the thing I want to highlight is the root data for now, as we're short of time. And essentially what we can see is that there is a similarity to the transcriptome's response to spaceflight to low pH stress on Earth and salt stress on Earth. This was absolutely fascinating. Initial hypothesis was that hypoxia would be the primary stress and be the most similar thing. So we started trying to think about what could have been increasing the acidity of the environment. It could be elevated CO2 and the altered convection occurrence on the International Space Station that could be contributing to this low pH stress, essentially causing the increased formation of carbonic acid near the root surface. It could also be a product of increased hypoxic environment. Essentially, anaerobic respiration causes the accumulation of ethyl alcohol and lactic acid. And if you have extra CO2 presence, that can change the flux through this particular pathway and potentially lead to more acidity accumulating. So a collaborator on the APEX-05 project, Wong Yu Choi, has recently went to the BEMA lab, and they recently published this paper where they looked at Arabidopsis thaliana under hypoxic environments. And what they showed was this increased acidity around the root system as they lowered the oxygen level. Essentially, they put these wild-type plants into an environment that had a noble gas instead of the normal atmospheric composition, and this induced hypoxia. And then you can see as a result of that, the dye that they had, this bromocoal purple, this pH reporter changed from this purple color to which you see in the air sample, to this like kind of yellowy color, which is indicating a decrease in pH as a result of this hypoxic stress. So ultimately, with this information in hand, we're then able to provide some recommendations to the NASA GeneLab and NASA researchers as a whole. Essentially, what we've learned is that the researchers need more time series data in order to allow metabolic flux analysis, not just in true microgravity, but also in microgravity simulators and atmospheric chambers on Earth. We also need more regular measurements of atmospheric composition on orbit, along with more radiation monitoring specific to each of the samples that are flying. Finally, it would be really helpful if there was a database of plant pictures that might help us identify physiological phenotypes to accompany these really awesome omics data. I really have to thank all of the awesome people that have come to the GeneLab Analysis Plant Working Group. And there's too many to list. Your usual suspects, you know who you are. It's a real honor to work with you guys, and I look forward to seeing you again in future missions, in future meetings. ultimately, finally, I'd just like to thank Dr. Sigrid Weinsard, because she's really been holding us together. It's like herding cats. So at that
I have to agree with that one, Richard.
So at that point, I would be happy to answer any questions. I might have rushed this because I'm a bit nervous, but because of the some scientists that are here.
We do have a question for you here. It says, can you please give more details about the bioinformatics pipeline used, and did you have to change the used parameters when applied on data from space?
The used parameters. Yeah, so the major feature that we had to change was actually the metadata. So in fact, when we went through the GeneLab metadata, one of the things we realized was all the different scientists that had been entering in the factor descriptions and the material types were doing it at different times and different places. Many of them were using different words to describe the same thing. So essentially, there was definitely a period when we had the analysis working group come together and go through these metadata and try to kind of curate it and tidy it. And we also realized that these factors by themselves are not enough. Essentially, we then created a metadata matrix that has 17 different major factors that need to be described in order to truly interpret the data that's coming out. That's now currently under peer review and will hopefully be out later this year.
Awesome. Thank you so much, Richard. Really appreciate your time and your talk. And our next speaker is Dr. Nicholas Brereton from University of Montreal. And today he will talk about the metagenomics at the murine host-gut microbiota interface during spaceflight. Take it away, Dr. Brereton.
Hi, can you see my screen there?
We can. We still see your slides, but yeah, perfect. There we go.
Fantastic. Thank you. Thank you for letting me talk today. So this is work coming out of the Microbes AWG, and as said there, I'll be talking about the gut microbiome of mice. We'll be using the GDLS 249. So this is data from the Rodent Research 6 experiment. So I'll just mention the experimental design or redesign of how we're tackling that data and the biological questions we're asking, and how then to take that data a little bit further by using some GeneLab resources. I'll then present some gut microbiome results and some results from the host gut microbiome interface, and just summarize at the end. So this is the experiment. So we've taken 32 mice, so a single cohort, and half of them were flown into space to the ISS, and half of them were kept on the ground as ground controls. They were then split again. So half of those mice in space were brought back down after 29 days in space as live animal return FLT, that is, and sacrificed with matched temporal ground controls, whereas the remaining half on the ISS of mice were kept there for 50 days and then sacrificed with matched ground controls as well. So we have 4 groups there of different treatments. So what questions are we asking here? So we're really asking, so does the spaceflight and habitation in the ISS alter the gut microbiome of these mice? And this encompasses ideas of short duration spaceflight effects, such as high gravity and the landing and stress of that, as well as longer duration spaceflight effects, such as microgravity and radiation, extreme built environment. The built environment's a whole field of research in microbiome science. You can see in the top right-hand corner here, the Harmony module, and environments like the Harmony module, such as surgical theatres, ICUs, submarines, which don't have great exchange with the environment and are cleaned repeatedly, end up being quite unique environments. They look a lot like the human gut a lot of the time. So are there effects? How do they affect the gut microbiome? We can also look at the samples we've got and how we compare them, and really think before looking at the data of what sort of questions we can ask and the different utility just beforehand. We're quite ignorant about what happens in the microbiome, but it's good to have a think about what we think could be happening. And so the cleanest question I think here would be the most extreme microgravity and radiation treatment, which would be the flight ISS versus the ground control. It's the longest microgravity experiment. We don't have that slightly confounding effect of landing and 1 gravity before sampling. We can compare the live animal return to their ground control. So it's a shorter microgravity treatment, and we do have those confounding other factors which make it a little harder to interpret. We can also add these samples together because we can be dynamic. It's GeneLab and ask different questions and do what we want with the data. So we can add the different spaceflight mice together and compare them to the ground controls together. So there's confounding there, but we get extra power because we get extra N. And that can potentially allow us to see some more subtle differences against the background of a lot of other variables to do with things like microgravity and radiation. So if you just look here, so this is bridging GeneLab data. If you look here on the right, this conceptual flow of how we really tackled going through this data, we can have some assumptions about what we think spaceflight is, how it's going to affect physiology of mice and humans really. And this builds upon great work done on the twin study and the cell paper looking at RNA-seq in mice. And we can expect that there'll be changes in lipid metabolism and steroid metabolism, glucose homeostasis, insulin resistance, immune responses, bone and muscle loss. And we can ask, do we think there's going to be gut microbiome interactions with these? And so we have fecal matter from the gut of these mice. We've done 16S, ITS, and whole genome shotgun here. And if we see differences, significant differences between spaceflight and ground controls, we can then go the next step to look at the interface between the gut microbiome and the host. So really looking at colon and liver samples, and we've got RNA-seq data from that, so gene expression. We can then take another step and look at the downstream effects to host phenotype by looking at host muscle, spleen, and thymus RNA-seq gene expression, which I won't talk about today, but is ongoing. So on to the results. So if I talk about, so just to start with 16S, so we use an approach called ANCHOR, which was developed for very high complexity samples, so soil and wastewater. And so each amplicon from 16S analysis has different utility to discriminate between different taxonomic levels. And so the data here, I know there's a lot of stats on the screen, but just singular take homes are, we identified ESVs could be identified as 31 species, 10 of these are ambiguous. So we had 100% identity of the amplicon compared to species, but it was common to multiple species. And we captured a lot of counts, so about 73% of the counts, a lot of the data at species level of these ESVs. So this is good enough resolution of these complex systems to ask some statistical questions of change. And you can see a lot of graphs here again. So on the left here, we've just got PCA. There's singular message to take away from here, which is if we compare our 4 groups, we can see they separate, the samples separate by those groups, by those treatments quite well, and by the unconstrained PCA at the top. And if we look directly at our pairwise comparisons of ISS versus flight versus ground control, or those LAR samples, so live animal return, ground control and flight, that all the samples separate, whether we constrain them or not by the question, quite well, and they're significantly different as well when we do the stats on that. If you look at these fold change graphs, we directly look at what's changing that differentiates these groups together, and we can see that there's more significant difference in microbiota in the LAR, so the live animal return. Remember, they've gone through that stressful landing and gravity after microgravity. But there are some commonalities between these groups. So they're one cohort, but in some ways they're independent. They've got different controls and they're separate groups. So Extobacter muris and Dicynobacter welbionis were both enriched, significantly enriched in spaceflight mice, regardless of if they're brought back home or not. And we also have some known unknown bacteria. So there's particularly two bacteria which we see in other studies but haven't been cultured yet, so we don't know what they are. And I always think it's useful to point them out. There's a lot of unknown there. So just to recap, there's something I didn't mention, which is that there's one bacteria which is very prevalent in these mice that didn't change substantially with spaceflight, which is Parabacteriales goldsteinii. But and that's the majority of the counts, but against the background of lower abundant bacteria, we saw significant change and substantial change. And it included these 2 bacteria, one of which is a butyrate producer. So that's short chain fatty acids and will interact with lipid metabolism, nutrient absorption, and is the resource for a lot of other microbiota in the gut. And we also have Extobacter muris, which is a really fascinating species because it's a real bottleneck functionality. So it's quite rare to see bottlenecks of function in really diverse microbiome environments, but it's similar to Clostridium scindens, if anyone knows human microbiome research, which it's a 7-alpha dehydroxylase producer and mediates bile acid production, so for primary to secondary bile acid metabolism. So because we have a change in this bacteria, we'd expect there to be changes in bile acid receptor interactions, altered bile acid pool, and changes in the liver. because 95% of those bile acids are recirculated back to the liver. And for those who don't know much about bile acids, they mediate the host microbiome metabolic interactions, so they're really important. So to go from that 16S to WGS, whole genome shotgun, Mike Lee and Emmanuel Gonzalez put together some wonderful WGS assemblies. We have 32 individual assemblies, as well as a single very large de novo assembly. And the WGS and 16S agree very well. You can see in the top right-hand corner here. So these two species of interest is that for the species identified with the 16S, they had on average over 99% sequence identity common with contigs and WGS. It's really rare to get different technologies that are this far away from each other to really match at the sequence level. So that's fantastic. But did we miss anything with 16S? 16S is really powerful for highly complex systems. There's a lot in the databases, and it's quite strong, but it's misses a lot as well. There's real big substantial biases. So I know the fonts here is very small, but chlamydia was missed, so the primers didn't pick it up. We see the parabacterioides, and I can get to species level with manual curation. We also saw viruses, archaea, plants, insects, and two very highly abundant protists and nematodes, so Plasmodium and Trichinella. And these, so early analysis, it needs to be criticized more, suggests these contigs with these are differentially abundant, so potentially responding to spaceflight. So to move on to we've seen some differences that are significant in the gut microbiome. So moving on to the interface between that gut microbiome and the host. So we'd expect here, before looking at the data, changes in the colon RNA-seq. So this is RNA-seq data, gene expression, pulled together by John Galazka really wonderfully. We'd expect immune responses, secondary bile acid responses, and changes in the liver of bile acid, particularly CYP7A1, which is a master regulator and is highly strictly regulated by bile acid, as well as knock-on effects to steroid metabolism, cholesterol, and lipids. Lipid metabolism. And so just I'll speed up a little bit, I'm sorry, but so this is a really busy slide, but there's only one take-home message again from this really, which is, so looking at the colon RNA-seq here, the gene expression, we did see steroidal hormone biosynthesis changes, immune changes, as well as metabolism of xenobiotics. And we also saw the Chagas pathway light up, and this is a pathway of parasite response. So doing a bit of Sherlocking here is, if we look at a Trichinella study where they challenged mice with Trichinella, there was 11 genes which were consistently differentially expressed. We have 9 of them being expressed in the system. John did some wonderful work of an extra little step of going a little bit off-piste and mapping some of the host RNA-seq from the colon epithelial layer to the Trichinella genome, and we see some nematodes, some Trichinella genes floating around mRNA, and that's cadherin tumor suppressor gene. So this is interrupting cell-cell adhesion at the epithelial layer. So what we're doing here is bridging different technologies together and bridging different specialties together, which is a great AWG strength. And just to mention in the liver as well, we did see significant difference in expression in CYP7A1, this critical point of regulation for bile acid, synthesis from cholesterol. So we imagine there's going to be a big change to the cholesterol pathway, and we do also see changes in steroid exporter and bile acid receptor targeted genes for cholesterol and lipid metabolism. And just to finish up, this is my summary slide. So we managed to identify some novel cross-kingdom spaceflight induced gut microbiome changes, as well as the fact that those changes were really coherent with spaceflight pathology. And we've observed direct host gut microbiome genetic interactions, and some of your ears might pick up there that one of the real challenges with this type of omic data is that we don't have a causal direction here. So we don't know whether there's a change in the microbiome that's caused change in the host or vice versa, or maybe we should be looking at this as one coherent system. And in general, that's the challenge of not really having highly tractable systems and highly replicated systems, which people are used to on Earth, editors, and sometimes it'd be a challenge to publish because it's used to very, very different systems that are highly tractable. We could do FMT normally to challenge these questions. But what we do have here is the strength of the AWGs to overcome that challenge, which is a lot of diversity of data and diversity of people and specialty. And so this leads to that priority for meaningful navigation of all this data and really working on dialogue between AWG members to overcome these challenges. So thank you very much. And do feel free to email me any questions as well later, or if you're interested in the work.
Wonderful. Thank you so much, Dr. Brereton. We do have a couple of questions for you here. First one is, are you using the same mice for microbiome 16S and feces and RNA-seq amongst organs?
The same mice? Yes. Yeah, so it's the RR6 experiment. So these are all coherent. The technologies make it quite hard to do quantitative direct comparison, but inherently we're dealing with the same biology. So we let the biology guide that analysis and guide querying that data.
Wonderful. Another question here is, have you seen any changes specifically in TMAO in the gut?
What's TMAO? Sorry?
Can somebody put that in the chat who wrote this particular question? What's TMAO?
If it's something obvious, I really apologize.
Okay, well, perhaps the person who asked this question, you can connect with, trimethylamine and oxide.
So keep in mind here, we also have I love metabolite data. So certainly in terms of the bile acids, you can really separate what the effects would be different in mice because there's some big bile acid differences, but we've also got hundreds of organisms there interacting across different kingdoms of life. So any extra information, I get the slight impression that there's not an incredible amount of metabolomics done, or I can't find it in a lot of data, and that would be a fantastic thing sort of to make some steps forward really for detailed analysis.
Wonderful. Thank you so much, Dr. Brereton.
Thank you.
Our third speaker in this category is Dr. Jean Agius from the University of Malta, and today she will present about extraterrestrial gynecology. Could spaceflight increase the risk of developing cancer in female astronauts? Take it away, Dr. Agius.
Thank you. So I'm just going to share the screen. Bear with me a minute because it just decided to disappear. And should be here. Yeah. Okay. There we go. I'm just going to make this smaller. And here we go. So first of all, thank you so much for inviting me to this really, really interesting symposium. So my name is Jean, and I come from the University of Malta. And basically by training, I'm a gynecologist. I've spent 20 years working in the clinical field. And then in the last 8 years or so, I've moved to university. So now I'm an academic. I'm the head of Department of Anatomy, and this is our university in Malta. Okay, so you might wonder, okay, how come from little Malta you've become interested in extraterrestrial gynecology and the link between spaceflight and the risk of gynae cancer in female astronauts? So basically, it goes back a number of years, pre-COVID era, when we're on holiday in Florida, supposedly to go and visit Disneyland, but my daughter here, Amelie, she got bored, and so did we. So we took the occasion and said, okay, why don't we spend a day at the Kennedy Space Center? And we're lucky to meet a female astronaut, Wendy Lawrence, and we're just so thrilled. In fact, my daughter wants to become an astronaut. So trying to bring the two worlds together, by saying we, I mean, I'm referring here, and I have to thank Afshin, who has already done a very nice introduction to our study. And we wanted to have a look to see whether there is any link between gynecological cancer and female astronauts. I mean, of course, so many advances are happening, and so much is known, and yet still, I mean, not everything is known, particularly related to female gynae cancer. So a little bit of background, which I'm sure you all know. So outer space is an extremely hostile environment for human life due to radiation and microgravity, amongst others. Spaceflight has also been shown to have an impact on established cancer hallmarks in general, and with the possibility of increasing carcinogenic risk. Now we know that terrestrially, women do have a higher incidence of radiation-induced cancers, I mean, the most well-known being breast cancer. And because of a number of reasons, I mean, females are permitted to spend significantly less time in space than men, been up till now, at least according to the guidelines of many space agencies. So the aim of our work and the aim of this presentation has been to focus on the effects of microgravity and radiation on the female reproductive system, particularly gynecological cancer, and to provide a summary of the research which has been carried out to date. Actually, there's not much related to gynecological cancer, and to highlight what further studies are needed to pave the way for safer exploration class missions, as well as post-flight screening and management of women astronauts following long-duration spaceflight. It has been shown, and this has already been published it's already known that radiation does affect female reproductive function. So this is thanks to the work of April Ronka, Lane Christiansen, John Stellar. There is, I mean, in all ages, it has been shown that there is the risk of amenorrhea, that is no periods in up to 50%, and sterility, whether temporary or permanent. And if you take women of reproductive age, so 15 to 40 years, again, I mean, there has been negative influence, negative impact on the reproductive function of females. Again, I mean, summarizing what is known to date on reproductive function. So I'm not just specifying on cancer here, but on the female reproductive system and the effect on the womb, effect on the ovaries. It has been shown that space radiation does lead to depleted ovarian follicles, and hence the sterility, and hence the premature menopause. In fact, that is leading to the high luteinizing hormone and FSH, and also the decrease in uterine size, which are all symptoms of menopause. And same for microgravity and hypergravity. There has also been shown to be detrimental on the female reproductive system. Of course, more is known on the animal rather than on the human. But on those rats, basically mice, which have been taken up in space and actually were pregnant, it was shown also that there is delayed development in the embryo in the blastocyst stage, and even in the birth weight of the pup. So this made us think, okay, if there is already this known, I mean, on the female reproductive system, what else could be happening on cancer? So given that there is not so much known on cancer of the female reproductive tract, the way we tackled this was to have a look at what is known so far on carcinogenesis in general, like irrespective of the part of the body. And again, although not a lot is known, but we did come up with, we did manage to retrieve some data. For example, on the lung, all right, so in lung cells, it has been shown that there are increased markers for DNA double-stranded breaks. There is reduced cell proliferation, persistent oxidative stress, increased colony formation, and that is under the effect of radiation. Also, there has been an increase in methylation, therefore suggesting a lasting impact on the epigenome relative, for example, to relevant for lung cancer. Also, in spleen, there has been upregulation of genes involved in DNA repair and cell cycle. Looking at other hematopoietic stem cells, also there has been, it has been shown that the linear energy transfer and irradiation has been causing early and higher incidence of tumorigenesis. And there was also a frequent occurrence of lymphomas. Intestinal cells, there has been a link with intestinal inflammatory disease and colon cancer, and also in the liver, increased incidence of hepatocellular carcinoma. And again, in the kidney, and there has been an increase in mutations, and also tests on human foreskin also suggest that there is a link with increased DNA damage and, of course, possible tumorigenesis. So that is what is known on space radiation cancer in general. And this is all found in a review, which actually mentioned previously. So this is what we know up till now, I mean, till a few weeks ago, effectively. So we decided to say, okay, let's look at the literature related to gynecological tissues and the effect of irradiation. And of course, I mean, there wasn't much known, there isn't much known yet. But from what we could find, there seems to be some conflicting information. Again, I mean, in this case, we're always talking of animal experiments, right? So mice. And in some of the studies, there seemed to be an induction of ovarian tumors, or at least in 2 studies. However, in the other 2, there didn't seem to be any increase. And actually, there could also be a correlation with inhibition of development of certain types of ovarian tumor, particularly the granulosa cell tumor. So conflicting evidence there. And if we look at the cervix, we could only come up with 1 study which shows the effect of cervical cancer and the radiation, which showed increased DNA damage in that 1 study, and same for the endometrium, the lining of the womb. There seemed to be a link, at least on experiments done on endometrial carcinoma cell lines. What about microgravity? So what do we know about microgravity and the effect on cancer in general? So looking at breast cancer, there seems to be a link with microgravity and breast cancer. So there were 3 studies that we came across. And so there were, for example, cells forming multicellular spheroids resembling epithelial ducts, and it also induced differential regulation of a number of cytokines and the number of genes producing certain cytokines. Same for brain tumors like gliomas. There was an induction of apoptosis and disruption of a number of signaling pathways. In the case of the lung, again, similar to breast, so there was upregulation of certain genes and formation of multicellular spheroids. Melanoma, and there was decreased cell viability and increase in caspase activity. Thyroid cancer, again, formation of multicellular spheroids and differential expression of a number of genes producing cytokines and proteins. Again, most of these were done on cell cultures, okay? Yes, we're coming to the conclusion. Again, we looked at what is known so far on the effect of microgravity and gyn cancer. And again, as expected, very little. And the little which is known is also showing conflicting evidence. So there were a couple of studies which showed that there could be an increase in spheroid formation. However, in one particular study, there was actually reduced proliferation, migration, and actually higher sensitivity of these cancer cells, of ovarian cancer cells, to cisplatin, which is one of the chemotherapeutic agents used in the treatment of ovarian cancer. Cervical, there seemed to be some correlation between microgravity and cervical, but again, we're talking of cell cultures here. And endometrial, again, in cell cultures, there seemed to be some kind of relation between microgravity and the effect on endometrial cancer. But again, very little, very few studies. And if you're interested, I mean, this is the paper which has just been accepted with very minor changes. So hopefully it will be out open access by the end of the week. And here I thank the 20 or so co-authors, many of whom came from the AWG network. So in conclusion, currently, therefore, there is no evidence to suggest that female astronauts do have an increased incidence of gyn cancers. However, there is scarce knowledge about the impact of space radiation and microgravity on gyn cancer, as there has not been sufficient numbers of female astronauts exposed to long duration, low dose rate, proton and heavy ion radiation to reliably determine the impact on the reproductive system. But with the upcoming missions, where there will be longer duration of exposures to both microgravity and space radiation, and hence the influence of flight length on risks related to gyn cancers, they will demand a larger focus in ensuring astronaut safety during flight and post-flight. Among all body tissues, the male and female gonads are among the most sensitive to radiation. So therefore, these tissues are best studied to determine space radiation-related exposures. Just like to end by inviting anyone who's interested to learn more about gyn cancer research to join this collaboration in science and technology. It's a EU-funded project called GynoCare, which I happen to be the chair. It actually looks at rare gyn cancer research, or let's say gyn cancer in, let's say, rare situations like in space travel. And you're very welcome to join. Basically, we've got over 135 members from 33 countries, from Europe and beyond. And our work is divided into work packages, which includes basic research, biobanking, legal and ethical issues, and also link with industry and pharma. So I'll end with saying thank you to at least these 20 people who were embarked on the exciting project of having our paper, our review paper published. And special thanks to Rosa and Ricardo, of course to Afshin, and Joe Borch for introducing me to the group. So thank you very much. And it was lovely being part of the symposium. Thank you.
Thank you so much, Dr. Aegis. That was really fascinating information and a great talk. We are a little bit behind, so I think we're going to go to the next speaker. But if anybody has questions, please feel free to put them in the chat, and perhaps Dr. Aegis, you can answer them.
With pleasure. Thank you.
Thank you. And our next speaker is Dr. Eric Antonsen from Baylor College of Medicine. And today he will speak about causal diagramming in human system risk management. Thank you so much for being here, and please take it away.
Okay, can you hear me okay?
We can hear you.
Okay, I am sharing my screen, and are you able to see it?
Yes.
Okay, fantastic. Thanks for the invitation to speak here. This is going to be a presentation that's pretty different from all the other ones that you've seen here. And the reason why is because it's about how we communicate and get the important information from all the work that all the scientists in this group do through to other parts of NASA as an agency. Everyone is probably familiar with these graphs, or at least some version of the risks that we use and the colors that we use to describe what we need more research and more understanding in. This one that you see right here is the most updated version that is available, meaning as of June 2022, this month. This changes all the time. And what I think many folks don't understand about this is that the way that it changes and who is responsible for those changes are a specific group at the Johnson Space Center called the Human System Risk Board. Very often, It is confused with the Human Research Program. The Human Research Program is one of the major stakeholders of the board, and they bring a critically important job of trying to fund the research that helps us understand these risks, and in many cases to build out the countermeasures that will help to reduce these risks. But the decision on whether these are red, yellow, or green and when they change is a responsibility of the Health and Medical Technical Authority at NASA. So one thing to know about these is that for the history of when they've existed, they have typically been studied in silos. So when I say a silo, what I'm drawing out there is taking one risk and showing that the risk itself across different missions that the agency considers. And the challenge with that is that a Mars mission is very different from something in low Earth orbit. And the reason why this matters is because the decisions on what goes into those vehicles, whether it's science or whether it's propellant for moving the vehicle around, or whether it's oxygen for the crews to breathe, those things all compete at NASA for space in these vehicles, habitats, and spacesuits that are unique to each of those missions. So when we talk about these risks, something like that sensory motor risk that I've highlighted is actually experienced across an integration of all these risks in a single mission. So when we talk about what's the importance of this or that risk, that they contribute and they affect each other. Everybody here is familiar with the idea that calcium homeostasis in the body is going to affect the bone fracture risk, and it's also going to affect the renal stone risk, for example. So these things are experienced in a rolled up way when crews go into space, and how we plan for that and how we make space for both science and crew health and performance system things are continuing and ongoing challenges for the agency. This information is available at the NASA Technical Reports server at the link that you see in the bottom right. And I'll have other reports that people can follow up on, because what we're going to talk about today is how are we trying to figure out how to draw those links between some of the basic science things that a lot of this community is doing and the outcomes in missions that the people who are responsible for saying what goes on Gateway, for example, are trying to wrestle with. So we use something called a DAG. It's a directed acyclic graph. It's a visual tool. It's a knowledge graph in a sense, and it's really a communication tool, but it holds the potential to do other things more broadly as we build them out. These graphs that I'm going to show you today have been built over the last 2.5 years by the entire human health and performance community that meets at the Human System Risk Board. And not just that, it means they've all had a chance to weigh in, to give feedback, and to argue about what should we put in these graphs. They're intended to represent at the highest level the best understanding we have of where risk comes from. And they're not intended to be static. They're intended to be dynamic, and they're intended to be updated as we learn more and get more evidence. And evidence from this community absolutely helps inform those things. So what is a DAG, right? What I'm showing you on the screen right now is a simple story that's drawn in a graph. And it starts over here with those hazards over on the left with the hazards of human spaceflight. And those hazards, for most of the risks that we carry, imply that something is changing about the human. And those changes, depending on how long you're exposed to the hazards, can modify the readiness of any individual crew member to go do the job we're asking them to do. That's the individual changes. But it's not just the individual who drives whether or not the job gets done, because each of those individuals are part of a team. They bring knowledge, skills, and ability. They bring different readiness levels, and they bring different responses to the spaceflight environment. And that team also involves the systems, intelligence systems that are put on the vehicle, as well as those groups that are back on the ground. And so that team can affect the overall capability of the crew, and that's a different part. Whether or not the job gets done doesn't just depend on the humans. If you didn't pack enough spares to repair the treadmill, then that's not getting fixed. And so the system itself also contributes to human performance. And whether or not you get those tasks done leads to some probability that you're going to lose mission outcomes. Now, this is not a formal DAG from the risk board. This is just a way to help illustrate telling the story that shows why it's valuable to get everybody on the same page. Each of these DAGs start with the spaceflight hazards, and end with a common set of defined mission level outcomes. These are the ways that we communicate with the program managers and the people who are responsible for making decisions about what goes on Gateway, what goes on a Mars mission, what goes on the lunar lander, for example. If science is going to make it on there, or if these things are going to influence this stuff, there is a pathway from what happened at the beginning of putting somebody in spaceflight to where is the risk. And what we've laid out for all of these over 2.5 years is a set of guidelines that set the standard for how we do these things in a consistent way so the whole community can use them. That is all available publicly right now at the NTS server at the link that you see at the bottom of this slide, and it'll be on some of the following slides as well. I wish I could talk more in depth, but I'm just going to highlight a couple of things. There are definitions for what gets included in these DAGs in terms of those nodes that I showed, and whether or not an edge, an arrow between the nodes, can be drawn between them. And those definitions are tied to a process for assessing a level of evidence. These things all get brought forward to the board, and they have to be approved by the larger community. In addition to that, those levels of evidence, there is a process behind them that is set up and defined by a number of the A. Bradford Hill criteria for causality that you see on the left, and a stacking approach algorithm that allows us to calculate or convey a level of evidence for a set of information, whether it's published evidence or unpublished spaceflight data from the astronauts, for example. We can say that the level of evidence that supports either the risk posture or these connections in a DAG ranges from speculative to strong. And so we've standardized a way of attempting to do that, and that's detailed within that within the Human System Risk Management Plan that's in the citation at the bottom of this slide. Of interest to this population, this group here, there are criteria that helps identify when animal and cellular studies would qualify to have higher than a weak level of evidence because of all the translational challenges that we struggle with in going from cellular, molecular, and animal data to humans. And so we lay out the standards and the criteria that we have for saying, hey, if you meet all these things and you show attention to experimental design and you also address the translational challenges and use molecular and cellular endpoints that are relevant to that translation, then these types of information and evidence brought from your groups can push up the level of evidence that supports either risk posture or these DAGs. So I'm going to walk you through, not an entire DAG because I just don't have the time, but I'm going to walk you through one of the risk DAGs that most people here might not be familiar with because it's one of the DAGs or one of the risks that is actually not funded by the HRP right now. So there are risks that we follow and track that are not currently active by the Human Research Program. In many cases, it's because we think we've found the answers and we think we know how to do things, but they remain risks because some of the stuff that's needed to keep the risk posture low may not make it onto missions in the future. So what you're seeing here, this bowl of spaghetti, is one of the DAGs. And I'm not going to go through the whole thing. I'm just going to highlight a couple of areas to help you understand how we talk this when we talk to program managers. So I've zoomed in to a little path tracing right here. Noise exposure that occurs on the ISS or in any other space mission, we used to call this the hearing loss DAG because the noise that occurs there can induce cochlear changes in the ear. And there's some probability that those changes will lead to a measurable hearing shift, meaning lower frequency or high frequency reduction in hearing probably, and then that will affect individual readiness that goes through and affects their ability to do their job. We called it the hearing risk for the longest time because this is what most people had in their minds about what's the risk, and it was a risk to health. But there's also a risk to performance that doesn't go through those hearing changes. That noise exposure can be something that just causes people to not be able to do their jobs, even though there's not a health implication. What do I mean by that? In 2003, the front page of the LA Times had the commander of the early ISS missions on the front page saying, I couldn't hear my crew and I couldn't hear what mission control were saying. So their ability to do their job may have nothing to do with whether or not they actually have hearing loss. It may just have to do with how we've engineered the system, the fans, the pumps, all the things that cause all that noise. And that's why there's an alternate pathway to getting to performance decrements. In addition to that, other risks can contribute. So the cochlear changes that we were concerned about that can affect health can come from things like ototoxins, for example. And these are two other human system risks that can contribute. So the toxicology risk, we currently monitor toluene, xylene, and the C5 to C9 alkanes on the space station that are known to have ototoxic properties. In addition, we have at least 23 medications in the ISS formulary that are known to have low to moderate ototoxic potential, 6 of them a little bit worse than the rest of them. But this is what I mean when I say drawing the connections between some of these risks and how they affect other risks. So we're attempting to map out what do we think we know about where risk comes from and how it builds through a system. Finally, there's a bunch of little lines highlighted here that talk about the system itself. How did we design it? What requirements did we have in place? What did we put into the system? What monitoring did we have in place that helps us understand pressure changes for the ear or those noise levels? Did we do pre-flight hearing status, and how can we detect hearing changes and tell us when we should be worried about using countermeasures that are in the system? So each of these DAGs has a high-level narrative that tells the basic story of what's going on here. And those are affiliated with those things. These are available within NASA right now. And soon, once we get through the processes, these will all be available external to NASA publicly as well. But the reason why this matters for you is that these are not intended to be the right answer. Right now, there is work going on the bone risk that looks at musculoskeletal loads, how they feed bone resorption and formation, how those affect density and structure, and how those impact skeletal fragility. A subset of this that a community like this can take and blow up into a lot more detail and understand when they've looked at that, how they can then feed it back into the rest of the risk pathway. So these are updates that go on at the board on an annual basis. They are intended to be dynamic. As evidence builds, they should be updated. The risk custodian teams at the Human System Risk Board are responsible for these, and the epidemiologist for each one is the primary point of contact. If you want more information about how to influence these things, these are the reports that are available on the public servers. And the Human System Risk Board and I can answer questions going forward. And just to give you a brief direction of where this is going, this is all 30 of those risks edge-worked and put together into one large network that we're exploring the possibility of structural and network analytics for trying to better characterize how these risks interact. So I will stop there, and I appreciate your attention. And if we have time for questions, I'm happy to answer them.
Wonderful, thank you. Maybe we have time for one or two here. So first question is, how can AWG members contribute to validate, maybe edit, and provide data towards the DAGs?
It's an excellent question. And one of the things that I did when I was still the chair of the Human System Risk Board was to ask and put into our management plan an ad hoc position for the space biology community to have a representative on the Human System Risk Board. In the past, that's been Lisa Carnell-Scott. Other folks may take that role. It's kind of up to the space biology community who that is, but there is formal representation pathway for this community to that board. So for whoever that representative is, the community should try to work through and with that representative. We've been working very closely with Ryan Scott and working on trying to identify pathways to help with the validation. And we welcome further collaboration to try to make sure that these are updated in a way that was consistent with the evidence. And you all are the owners of that evidence and the best people to interpret it. So please help us.
Wonderful, thank you. And apologies for the audience, but we are running a little behind, so we do have to move on to our next speaker. And our next speaker of the event is Dr. Masafumi Muratani from the University of Tsukuba. He will be presenting about space life science research to predict the future of life on and beyond Earth. Thank you for joining us. I know it's very early for you in Japan, but go ahead and take it away, please.
Thank you very much. Actually, it's a very good time in the morning time. So thank you very much for giving me opportunity to talk about this space life science research to predict the future of life on and beyond Earth. So I'm helping the genomics analysis for JAXA project. I also did the human astronaut liquid biopsy study as a PI. So far, like there are multiple projects, the mouse flight and also human flight. And in parallel, there is a GeneLab huge data sets, and also astronaut human study, the twin study here. Which recently published many papers, but I think we are commonly aiming towards the understanding of the environment response in space by integrating the human, mouse, and other animal, plants, other sort of data, and that's really like a GeneLab type analysis, I think. So in my project, the liquid biopsy is used. This is just a cartoon showing this liquid biopsy is very useful to monitor what's happening in the inside of the body, like a cell death or secretion from the cells, RNA, DNA, and other biomolecules are running in included in a extracellular vesicle. This is very useful because this is a less invasive monitoring method for internal tissue. We can, of course, use in mouse and human. Mouse is a very great model because we can harvest lots of tissue directly and genetically identical, relatively large N number and artificial gravity control, which I will explain in a minute. And also in human, it's of course, it's important model and time course sampling. And for liquid biopsy, it's very advantageous to get a lot of blood actually. From mouse, it's a max like a few hundred microliter. So this is a brief introduction of the cell-free epigenome study. This is a 6 astronaut study, liquid biopsy study. We collected baseline, in-flight, and then post-flight time points from 6 astronauts, and we performed lots of RNA-seq and DNA sequencing related assays. And I don't have enough time to explain the detail yet, but this is just a PCA plot of the cell-free RNA analysis. As you can see here, the pre-flight 6 astronauts time points are quite scattered, but once they go into the flight, it's packed into this section, and post-flight packed into this section. So the 6 astronauts all the time point are behaving very nicely in a consistent manner. This is a very good news because we are wondering, like a 6 astronaut study, clinical study, if we can get a nice result or not. And this is a very brief kind of like summary of what we can see in altered cell-free RNA in space flight and pathway analysis by Metascape. So we can see, firstly, we can see some different pathways like oxidative phosphorylation, regulatory the vitamin D pathway, and like some brain or behavior related genes. And not just blood or immune related the pathways, but we can really assess what's going on in the central nervous system or inside of the body. This is tissue specificity pathway analysis, like a cerebellum, skeletal muscle, and here are some other tissues. And the question is now, how about in mice? JAXA has this MARS, Multiple Artificial Gravity Research System in Kibo unit. This is a really great system to assess the microgravity effect in space, which has a rotating platform, and by comparing this rotating situation and stable rotating platform, we can actually compare the 1G onboard control and real microgravity in space. JAXA have been performing multiple mouse flight experiments here, and I have been participating in this some of these flight experiments to provide the genomics analysis, like RNA-seq, ChIP-seq. This is a video from the publication from JAXA paper, like a mouse in 1G control. The mice are nicely living on with ground base. And here it's a micro Z situation. It's just like astronauts, mouse are flying around like this. but they are eating and drinking nicely, and healthy mice came back to the Earth. And this is a liquid biopsy again from the mice study, ground control. Actually, ground control is harvested at this timing, but 1G control, microgravity, and then time course adjusted ground control. Briefly, so the point is that we can compare artificial 1G to microgravity and ground control to other conditions to check the general flight stress and gravity effect. And here we can see the Venn diagram. In human, this is a human disease pathway enrichment analysis disease term. And here is a mouse gravity comparison. And actually, half of this mouse term were reproduced in human, which is impact of altered gravity. And this is a word cloud to show the kind of tendency of this visual, mitochondria, muscle, optic, atrophy, cardiac. So this process might be affected. And now, by collecting the human data, this is really the time. We have full set of mouse data and human data. Then what can you say? So current idea from us is a kind of conserved gravity-linked space phenotypes is not just mechanical unloading. And in mouse, human, and also some zebrafish or medaka fish analysis, skeletal muscle and bone change, of course, as expected happening. But even this water, living under the water, fish has a bone change. So this also indicates this is not really the mechanical unloading situation, but we can also see seemingly unrelated responses like oxidative stress. Muscle is just not mass, but fiber types, immune system, circadian rhythm. And in addition, we can also observe the intergenerational effects on DNA damage response and so on, actually. And based on this phenotype, we eventually came up with some idea. Maybe these changes might be linked or very similar, actually, to the functions gained during the sea to land migration. So from the sea to land, there is a quite important change happened for homeostasis, like a mineral balance, muscle bonds, and also the vestibular system and other types of systems, and also the environmental stress. They are living in the air, up on the ground, so high oxygen compared to in the sea, UV damage and skin dehydration or protection or sensation. These might be, we consider this is a kind of throwback phenotype. Once the vertebrates go into the space, these important 4 million years of evolutionary effort, really the effort, appear to be suddenly lost in space. So based on that idea, so how can we explain? Our working model is like this. So hidden adaptation stabilizing the ubiquitous environmental signal. So this is the gravity on the Earth. In the creature on the ground, they never lost gravity evolutionary. So here is a apparent evolutionary traits, kind of graph. And then we tend to think this is all evolution, but there might be a significant portion of adaptation, which is the less stable evolution here. And then on top of that, good portion of adaptation. And this is robustly maintained by gravity potentially, and also inherited by intergenerational transmission. And then once the vertebrates go into the space, their adaptation pinched by, locked by the gravity is suddenly lost and go into the naive state of vertebrate in space and appearance of the ancient phenotype. And this model is actually useful once we consider this huge portion of adaptation, that we can vectorize the environmental pressure and adaptation rate. That also indicates we can actually model in the future change by training the model and then simulating the future situation. even for the evolutionary unexperienced environment, like Moon and Mars situation. And also, this model is potentially useful to explain some of the important questions in evolution. So here, kind of sudden, like going back to the old phenotype, the Haeckel's biogenic law, for example, this can be explained by just the loss of this adaptation portion or masking during the development, and so on. And for some people, this representation might be more understandable. Here is the stability of the status and regulatory landscape. And then current phenotype is stripped into the ancient phenotype by loss of this adaptation target. And there could be many views. So we set up the open science topical program, Space Adaptation and Evolution. And our current member from Japan includes these experts from the anatomy and development and so on. And we also asked the international collaborations. So currently we are always inviting affiliated research projects, collaborators, and sponsors through AWG. So thank you. This is my final slide and acknowledgement. Thank you very much for your attention, and I'm happy to take questions.
Thank you so much, Fumi. That was an awesome presentation. We are right at time here. If there's any questions, we can take one question perhaps. But otherwise, I would like to thank KBR for providing the platform and hosting this event. And I'd like to thank Marco for the stellar support. All of the speakers for taking the time out of their busy schedules to tell us about their science. I thank you. And lastly, but not least, the audience for joining us and listening to some of this awesome science and joining the AWGs. So that concludes this year's AWG Symposium, and keep an eye out for future ones. I hope to see you at the next AWG Symposium. Thank you so much, everybody.